السلام عليكم ورحمة الله وبركاته. أعوذ بالله من الشيطان الرجيم. ورب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقه قولي. SubhanAllah al-Azim. We're reaching the conclusion of BIOC17 Microbiology, 10, class 2. So inshallah ta'ala, need a finale. Good morning, how are you? Doing great, how are you? You did it before. Yes. One just final notification about incubators. Should I still use the top left? Yeah, wait. Oh, I was thinking, oh, never mind. Oh, it sounds too good. Yeah, to hear. But apart from that, not really. I don't think I have questions. If you don't mind, their biofilm, that kind of stuff. Yeah, but that's consolidated to two different docs. Okay, that's okay. Thank you so much. No. So inshallah ta'ala, students are going to look back into the past, review material from weeks 1 to 10, inshallah. At least, if not this week, maybe just to give you a rundown, bid'Allah ta'ala. They'll be interpreting their differential media, they'll look back into their TDT results, inshallah ta'ala. There'll be a few things that they'll be hopefully looking forward to. Inshallah ta'ala. Also making mention of the final exam, or inshallah ta'ala to have it for essentially the time being that to go over thermal death time or the D values and across temps, but actually importantly talking about the different medias, right? Just a recap and also reflective practice that will connect the dots hopefully inshallah ta'ala. But I'm just gonna go back. So essentially interpreting the data, looking at a lysozyme assay, standard curve, and trying to interpolate, the different dilutions that are shown here. And to my understanding, these are from 2 different samples, the ones that students will see as examples across dilution factors. So inshallah ta'ala, I hope students will understand the material, will be a reflective practice. I hope inshallah ta'ala will connect the dots to what we've learned. And inshallah ta'ala, I look forward to having a brief review seminar, 2 seminars next week, going over labs 1 to 5 as a review for about 3 hours, 2 hours and a half or so. And then the latter half, and that's planned for Tuesday, next Tuesday, inshallah. And then the following one for the following Monday, bid'Allah ta'ala, from 10 to 1 as well. And that will focus on the remaining labs from lab 6 to 10, including this week, inshallah ta'ala. So with this, may Allah subhanahu wa ta'ala make it easy for the students. May Allah subhanahu wa ta'ala accept all of our efforts. And subhanAllah, our golden lab is coming to an end if Allah wills, but inshallah ta'ala, I hope down the road it will continue with a legacy within and beyond our efforts, or beyond our golden age, or even beyond these 2 courses of AO2, Introductory Biology, and the Microbiology course. May Allah subhanahu wa ta'ala accept all of our efforts for these 4 courses. SubhanAllah, we're reaching, nearly the 35th seminar, 34th right now. And SubhanAllah, it's only by the mercy of Allah subhanahu wa ta'ala that hearts find rest, hearts find comfort, and minds find ease, knowing that all of this is from Him, Jalla Jalaluhu. So with this, may Allah subhanahu wa ta'ala make it easy and allow us to best please Him, serve Him, and be among souls that show the best of excellency beyond bounds, but beyond dimensions, beyond but beyond one's imagination. And may Allah grant us the best in this life and in the next. May Allah grant us Jannatul Firdaus. May Allah forgive me for my shortcomings and rectify them. Allow me to change completely to the day I meet Him, best pleased with me out of all of His creation. Allahumma Ameen. Wa akhiru da'wana alhamdulillah wa ameen. Barakallahu feekum. Inshallah ta'ala. Now we conclude briefly the Fatih. Like mini synopsis or forward. But inshallah, all of us will. May Allah subhanahu wa ta'ala grant us the best. All 8 gates of Jannah to Firdaus, or of heaven, Jannah for Jannah to Firdaus for us all. And Allahumma. Do you mind, Garvin? Or how's everything? If you don't mind, maybe once you're done, if you could wait outside, I'm just gonna fix it. Okay, I appreciate it.
Like if you like a range that's the thing. Tuesday. Yes. Lab performance in
That's one of our G2. It's in the back left corner, by the way. In the-
Something that's not over here. So are you your files from there?
Yeah.
Yeah. That's a big.
Oh, yeah. You want some of the bigger captions you want?
Sorry, yeah, it is. But so I wasn't that I found voices or thorough question and I got quite a lot. And then I have a question. I'm But I kind of like it because it's scary when I'm talking. You're not actually when you look at the car window.
It's a cold. It's cold. Get the same bottom. So she said.
It is. It's easy to like the edge. You always see what's. This is.
Yeah. I will like
Based on how nice faces that they're making sound. Like they choose this one. The last one. Oh, like it's okay. I just straight them like that.
yeah.
I thought we were racing. At one. Yeah. He's so rigorous. Did you see what? Yeah. No. Yeah, I think it's like a long hour, because then I can probably go examine. I don't know where I was at. I think like maybe between very long, I would like.
Hello, everyone. I hope you're all doing well. We're in our final in-person lab. Very tense indeed. Tense because I'm gonna miss you guys. Perhaps, yeah, so bear that in mind. Of course, if you have questions, any thoughts, complaints, concerns, points of compliments, considerations for the future. If you have any questions about grad school, med school, let me know accordingly. Now, a few important things that I just want to highlight before I move further. One is that I have, let's say, two sides of a coin where essentially I try to be both just in the sense that there's certain guidelines that I expect that I've shared through Quirkus. Sometimes they are either above or below certain expectations that maybe Dr. Pun shares, because essentially I, after taking this course, know, and from other courses as well, what it means to be scientifically acute. And so that's why I want to make sure that you guys, independent of your journeys and your aspirations, are able to essentially meet certain guidelines in place. That said, often what makes me distinguished from others, and it's not to boast in any way or shape or form, is to highlight the mercy aspect. Many other TAs won't scale grades, right? And I do this because I want to account for any level of variability, any inconsistencies, miscommunication, things that maybe weren't understood by yourselves or by myself for specific reasons. And so those are reasons that you probably would have noticed the scaling of the FLR, the first one. and likely for the generation time, which if you don't have the generation time assignment or the unknown, please come by to me because I don't want to have to keep, I want you guys to actually have your materials, the ones that you, after all, worked on. So bear that in mind. A few other reminders that you guys actually did. Some of you actually, generally speaking, generation time assignment was a well done, really amazing. The average was an 87, which actually even unscaled is exceptional for our practicals. So well done. This is both practicals combined, so it's a really high score. Some of the comments that I've shared includes essentially using a slope, because essentially some of you may have drawn curves that are round for your generation time assignment, and pretty much I'm going to try to draw that out. Maybe part of it. Idea is that, let's say this is your curve. Different curves for different assays have different formats. So like the lysozyme assay, which you're going to see the following page, is curved. We'll see it afterwards, but the point is that for like thermal death time, for the generation time assignment, you have them in linear, like you have a line of S. So your points, be it from the microplate reader or from viable counts, in either way, should essentially follow through a line of best fit. If you haven't done so, then the issue mathematically, or using essentially certain rules, would essentially not follow through with your most reliable slope, because if you use a curve, there could be multiple tangents, multiple slopes that you work with. So you thereby want to make things consistent, especially with semi-long paper. Generally speaking, you're dealing with a line of best fit that's usually linear as a curve. So just bear that in mind. For the lysozyme assay, which we're going to see soon, your curves are, in fact, there are different terms for this, but essentially the standard curve is essentially is nonlinear. So just bear those points in mind, especially for your lab exam, because you want to bear in mind what you expect as you plot out certain values. So keep those details in mind. Any questions? No questions? Okay, so let me just make sure there's a few other things. So for the FLR, there seems to be a bit of miscommunication. And maybe I'll just highlight this for you guys as well. I mentioned about the tables, right? I expect, like, based on my Crocus inbox messages, and hopefully everyone has seen them. Please raise your hand if you've not seen my messages, because I do want to make sure that you guys are prepped for other things in store. Pretty much what I expect is, actually, let's go. Do I have time? No, I don't have time. Okay, so just to give you guys an understanding, yes, the deadline, Dr. Bun mentioned that you won't get a penalty until tomorrow, i.e., sorry, today. She said that tomorrow, meaning that it would be today, 10 p.m. However, the thing is, on Quercus, I don't know if you guys will actually be shown a penalty mark. So until Dr. Bun clarifies, then I can't say anything. Essentially, those are her words, but essentially, I recommend, if you've already essentially submitted the FLR, that's great. You don't have to worry. Does it? It's updated? Okay. So if that's the case for you guys, then that's great. And I actually personally so I had like a mini meeting for you guys prepped yesterday. If anyone was waiting for any reason, please let me know. But I saved that meeting for you guys because, unlike other practicals, they were able to have a final meeting with their TA to discuss certain requirements, expectations for that FLR. So pretty much that would subsidize for that point. But essentially, if you have final questions, let me know. Likely near the end of the practical or via email is also preferred. Let's see other things. Notebooks. So yes, in terms of the quality and your attendance for notebooks, I have like a checklist of people that have essentially have shown me their lab notebooks punctually and also with the expectations of the post and pre-labs. So if I don't have you written down, please let me know. I believe for the pre-labs of week 8, I have 3 folks. That is Zara, Amro, and Cynthia. So we'll talk about that in due time. Like if I mentioned your name, you could come by and I could check your notebooks. And then those that I've mentioned today early on about certain things that they need to complete for their pre-lab, intro, post-lab results for UV, for the thermal death time, the environmental conditions, pH salinity, and osmolarity. Complete those points, and inshallah, by the end of today, you can be able to show it to me, and I could just check you off. See, the thing is for me, I understand student life. I understand that you guys may, some of you come 2 hours away, just to come to this lab, or maybe, to fulfill your academic obligations. But this is why I want to be generous, giving you guys the opportunity to come back, correct certain mistakes, right? I want to make sure that you guys have an amazing experience in this lab, and I will do my best to deliver that. If you feel like I haven't or I'm shortcoming, please let me know in person, email, I don't mind. But I just want to let you guys know that I'm trying to do my very best, independent of what reality seems to be. So that's pretty much my final point. Any questions? Any questions? Questions? No? Okay. No questions? Okay, let's go straight ahead to the lysozyme assay. That's your last practicals, well, at least the last section, last lab section in the lab manual, apart from the appendix, which you want to bear in mind, appendices or appendices A to D. But the lysozyme assay is one of your final in-person exercises, which, by the way, is virtual because essentially this would have been done from weeks 10 to 11. But the idea is that it's a special enzyme that is found in tears, sweat, saliva, and different salivary glands. So actually, even though it's not mentioned here, for those that are interested in physiology, you have special glands, those that are essentially around the maxillary glands, salivary glands, and different tissues, even mucous membranes surrounding them, like within the respiratory tract. You have lysozyme, right? And in the digestive system, it's important because that's where you get substrates broken down into chyme, right? So lysozyme is an enzyme that breaks down certain chemical bonds, and it's also noteworthy in relation to microbiology because it breaks down the outer cell wall of bacteria. Now, essentially, what you would have done, or what would be shown here for part A, is essentially creating multiple tubes. So you're creating a list, or you're preparing a list of different tubes based on the different conditions shown here. And likely you would have started off with a stock, like a lysozyme stock, which is interesting because you could get lysozyme from your own saliva. So just imagine like the relationships, which you'll see in a few slides away. But the idea is that, and hopefully, Jacob, let me know if you could see the screen. I'll just zoom in if needed. You can see it. Sorry, Hajra as well. I have some students that are at the far end, so I want to make sure they can see it from this end. But the idea is that you see how the stock increases, and this is so amazing because it connects me back to AO2, because students also essentially look at different stocks, but for NaCl on the growth of plants like duckweed, lemnas. So essentially, it's an aquatic bioassay. And essentially, the idea is that you are changing the concentration of your enzyme, which is shown here, right, from tubes A to F. And then G is essentially your bacteria, which is consistently 2 mils, except for the blank. And that bacteria could be E. coli, it could be Staphylococcus epidermidis, and you would essentially compare using the spectrophotometer the OD readings, the turbidity readings, because essentially, as you increase in lysozyme concentration, you're increasing in the breakdown of your substrate, which could be, in this case, bacteria, and thereby reducing the turbidity over increasing concentrations of lysozyme. Does that make sense? Anyone want me to repeat? No? Okay. So this is where actually the standard curve where it comes from, because essentially as you increase in the concentration of your lysozyme, which by the way, I would like you guys to be specific on. So you could say lysozyme concentration, brackets, nanograms per mil, close bracket. And then y-axis is your OD. It shows here at 450 nanometers, which is something you want to keep in mind, the difference in wavelength, unlike our other ones. But the idea is that as you increase once again in lysozyme concentration from A to F, tubes A to F, you should see a decrease, or you should see an increase in breakdown, increase in lysis of bacteria, and thereby a decrease in turbidity over increases or over essentially lysozyme concentrations increasing. You have that, and that's what you would see. And what you actually expected to do for this lab and consider it part of your post lab is to essentially interpolate. And this is what I mentioned on Quercus as well, but try to interpolate these values or draw out the standard curve shown here. And it could be a sketch, right? It's in normal grid paper or in your regular like in your notebooks. You could just draw out a grid like with these simple increments and then just go down exponentially in value, or at least the curve itself will go down exponentially with regular intervals of 500 nanograms per mil. So those values will go up in nanograms as the OD readings go down. And then what you're expected to do actually is to account for the different OD readings that are shown here from two different samples across two different dilution factors. So let's do an exercise. Let's say I'm looking at this value here. This is the OD reading of patient 1, right? Or let's say you're in a clinic, right? And you want to test the concentration of lysozyme, perhaps to see, like in connection to the Snyder test, how much bacteria, i.e. lactobacilli, could be broken down through lysozyme. And based on that, you would see the turbidity readings. So let's have an understanding about this. You have the turbidity readings of bacteria over increasing lysozyme concentrations. So let's say I go to here, 0.43. I'm going to just show it as an example. I'll show it with the laser pointer. So let's say 0.43 is around here. Back in my time when I was like an undergrad, I'd be very specific. I'll try to actually get like 86% because that's 0.43 right there. So let's imagine that's 0.43. I'm going down, going towards the curve. And then I go down from the curve onto the x-axis, right? So I went from the y-axis all the way down to about here in the x-axis, going down from the curve. So down the curve from the y-axis to the curve to the x-axis, we're at about 623 nanograms. So that's 623 nanograms. And, you know, like, I know you guys may be smiling. How do you know that? So essentially, like this is an estimation, that's correct. But at the same time, I'm also very peculiar. So I kind of take these values into account. And so let's say this is 623 nanograms, for example. So then what's next? Is that your ending value? What do you guys think? Say yes or no out loud. Is that your concentration of lysozyme in saliva? I heard some no's. Can I hear from everyone else? Yes or no? No. Why? You can raise your hand or you could say it out loud. Correct. So kind of got it, right? So what you would do then is essentially, actually, I want you guys to tell me, what would you do? 623 nanograms, what do you do to get the original concentration? Multiply by 10, multiply by 10, multiply by 10, 100, 1000, 0.1. What are you multiplying by? Multiplying by 10, that's right. Technically, you're dividing by 10 times, so you're technically dividing by this value here, which is multiplying by 10 to the power of 1. So that's essentially multiplying by 10. So you would get essentially 6,230 nanograms, or more precisely, you could say 6.23 micrograms per mil. So those are values that you want to just bear in mind for your lab exam. If units are specified, right, to convert, just make sure that you know how to do so. One microgram is 1,000 nanograms. 1 milligram is 1,000 micrograms. So just bear those units in mind. The difference between milli to micro to nano, and pico and femto are another ones. So if you're asked about it, just let me know. Or if you have extra questions, yeah. Hey, I don't have access to it, so I wouldn't necessarily know, but expect to do so actually, right? Expect like because you have many. We'll talk about this actually at the end. Remind me if I forget. But please, folks, do the same for the other values. So you actually want to do this in your lab notebook, and I do want to see it actually. So try to do this very simple, probably 5 minutes to determine after you draw out the curve. So try to do that as soon as you can. And this experiment would be done both at 1 minute and 3 minutes post incubation with lysozyme. So I want you to think, do you think that curve would be like comparing, let's say this was at 1 minute with lysozyme included in your bacterial incubation. Do you expect it at 3 minutes to increase, decrease in slope, or change or not change? What do you expect? Anyone? Feel free to say out loud. Do you think, like, let's say this was at 1 minute with lysozyme included, how would it change after 3 minutes of incubation with lysozyme? Increase? Decrease? In what way? Let me just draw it out. Imagine this is your lab exam. You have a curve that looks like this. You have a curve that looks like this. And often you're given like another one, which is like to see if you guys understand the material. So let's say this is A, this is B, this is C, and this is D. After 3 minutes, so C is technically what it would appear at 1 minute. At 3 minutes, do you expect it to be the purple one, the blue one, the one as before, so unchanged, or green? What do you say, Carlos? Green? Everyone say green? Does everyone say green? Raise your hand if you think it's green. Okay, raise your hand if you think it's blue. Blue? I'm seeing some people not raise hands. Actually, I want to see you guys raise your hands. Feel confident. I'm not marking. I just want to see what you guys think. The answer is green. So the reason is, indeed, actually, you expect at 3 minutes to be greater, right, incubation with your substrate, your bacteria, and so that'll be greater lysis of your bacteria over time. So this, by the way, connects to biochem. So maybe if you guys want to think about the connections, we could see the relationships in due time. Any questions before I move further? Don't worry about this because that's pretty much what I just mentioned here. The idea is that you want to be able to determine the original concentrations, right, of lysozyme in undiluted samples. Yes.
So you want to know how to like stack up, let's say.
That's correct. Remove, like, you don't have to do these other ones, but just that one. I'll see if I can clear it out for you guys. Yeah, so you want to draw this out in your notebook. And then you want to plot out or interpolate what these other values could be, or what the concentrations would be of the diluted, and then calculate for the undiluted sample, i.e. your stock of saliva, or stock of lysozyme in your saliva. And that would be for patient 1 here. This is patient 1. It's a bit hard to draw, but this is patient 1. And then we have patient 2 underneath. And you want to do it for both dilutions, so 1 and 2. I want to answer, Marwa, is that also your question, or is it a separate one? I saw your hand raised. Well, one moment. Did you have a question? Yes or no? Yes. Yes, you can show it to me afterwards. But yes, Mohammed.
You know how it's like very research the opposite also
Yes.
or like 30 seconds or
Yeah, less secret. I want you guys to think like this. Think about extensions. Not like, I'm not like, by the way, folks, I'm not favoring people. I'm just saying that I want you guys to think critically. Think about like, okay, if it was the other way around, let's say 30 seconds, then perhaps it would appear like this. Or what happens after 3 minutes and you find it to appear like this? What could have happened? Maybe cross-contamination, right? Perhaps additional bacteria were inputted into your cuvette, or it could be the case that you were rubbing the bottom of your cuvette, so the absorbance readings appear, overestimated. You want to think about that. Another thing that we didn't talk about is blanking, because if you look back, you notice that your first column is with your blank. So technically speaking, always expect for your lab exam for other like practicals that you normally want to blank, just like how you tear your mass balance, you want to blank the absorbance readings for your spec 20, for even the microplate reader, right? As we looked back to the TDT results, which we'll talk about soon. But I want you guys to think critically, because these are questions that, let's say I didn't say, let's say you only looked at what I talked about. Like, oh, that 3 minutes, so it looks down, like it goes down further. Then what happens if Dr. Brent poses you the same question about it being at 30 seconds, right? So you want to bear those critical questions in mind. A lot of it reflects back to the reflective practice, critical discussion questions in the lab manual, on the slides. So try to prep well for your lab exam in that way. Any questions? See you. Any questions? Any questions? No questions? And folks, I am paying attention, so please bear in mind that if you are talking, if you're on the phone, I do see this, so just bear that in mind. Okay, it's reminded me something important, and that is your code. Each of you have a code. C1, C2, C1, indeed. So I have a spectrum, if you recall, from 60 to 100% of what your lab performance is, right? Now, A was, so I'm going to cut it short. Essentially, don't be stressed. Don't be stressed, folks. C represents, so the letter C represents a grade between 80 to 90, and D reflects a grade from 90 to 100. If you have a C1, that is your quadrant of like the first 5%, i.e. you're somewhere between an 80 and an 85. If you're C2, you're somewhere between an 86 to a 90. If you're a D1, you're somewhere between a 90 and a 95. If you're a D2, you're somewhere between a 96 and 100. I want you guys to, digest that a bit. I also want to say, very important, I expect you guys to keep your quotes to yourself, just like how I give you guys like your grades. Don't share your grades with people because one, you don't want to make others feel bad if they get lower grades, or two, importantly, that's not the purpose of essentially, there's a concern that was brought up that I want to make sure you guys understand. I try to be, once again, just with you guys, right? So bear in mind that if, let's say, you showcase this week and for next week, inshallah, with your review seminar, an increase in participation, an increase in proficiency, an increase in performance during this last in-person lab, and then virtually with participation thereafter, then your grade can increase hypothetically to another quadrant, i.e. from a C1 to a C2, a C2 to a D1, a D1 to a D2. Or the other option is that within your grade, you could still go up from let's say a 90 to a 95, right? So I want you guys to think about that. Think about how you can improve based on what I've suggested already. And as we complete the remaining portions of today's lab, looking back at the differential media results, looking at the biofilm, talking about TDT, and there's one more. The lysozyme assay. Once you guys show these results, describe them, place them into your lab notebook, show them to me, show a level of engagement if you guys want to increase in your level, but also bear in mind that don't put hardship on the TA to expect that your grade is going to now be 100%. I want to recognize that everyone can get 100% without question, right? I'm not saying it's impossible, and I'll never say that. But the goal is to understand that you have to put the effort in. I try to put the effort in ways that perhaps, not to compare, but in ways that probably other TAs won't, right? So I want to make sure that you guys have ample opportunity to deliver and showcase what your performance is in the lab. So please bear that in mind. That includes our review seminar upcoming, but even from the past when it was, the snowstorm, taking the time to do a review with you guys for lab 6. So I want you guys to not feel any way, overburdened or at worst, uncompensated for, any inconveniences within the lab. So just bear those details in mind. And yes, I would appreciate that you guys consider where you are right now and how you can improve if needed or if you can. So bear those points in mind. I'm going to give you guys the media in just a moment. If in the meantime you have questions for the UTFR, I can answer them as I select or as I give you guys your book. You can say them out loud. I don't mind.
Are these grades for the 16%, like of the participation in the lab notebook, or just 8%?
That's a great question. So to answer this question, that reflects the 8%. Meaning, for your lab notebook, and this is why I just want to stress, if any of you have missed any components that I mentioned from the past, include your figure caption, your table caption, being descriptive, being noteworthy, note taking, your results, the discussion, and the discussion form includes not just the question right on the slide, it includes the incorporation of your observation. It includes essentially the result that you're able, I think I gave, this was, so just bear in mind that, I want you guys to be able to deliver, I forgot what was the last point I said. Yes, the lab notebook. If you're missing any outstanding items, and if you've made corrections from the past that you haven't shown me, this is your time to do so, right? We do have a bit of time. We're at 10 o'clock, I believe. So we'll have a bit of time to go over your differential media. Let's spend about 5 minutes. Sorry. Okay, maybe a bit more because you guys will actually need to draw it out in your lab notebook. So, okay, so within your lab notebooks, this is considered your results. Please have detailed figure captions. You have the MSA Mannitol Salt Agar plate. You have the McConkey Agar plate. You have the DNase DNA hydrolysis plate, and we'll look at the gelatinase test plate, just test tubes shortly. But I want you guys to look at your results, see what you observe, and tell me what you guys observed actually. I'm gonna open the slides for lab 9 so you guys could also see the results accordingly or the expectations. So let's talk about the Mannitol Salt Agar plate. Have a look at it now as a group. It's, the reddish pink plate. And based on your quadrants, i.e. 3 sections of the control, the 2 bacteria, I want you to tell me what you observed. Do you see any yellow colors? Do you see everything red or pink? Let me know what you guys see accordingly. I don't think they're really cool, but apart from that, sorry, did not access those. Apart from that, that's okay. That's okay. Great job. No problem. Give you yours. Just give me 1 minute. Yours is. Okay. So what do we see? Can I get a raise of hands if you see a yellow color? Or a yellow tint to your MSA plate? None of you? Really?
Like a home. And a yellow color.
It is yellow.
This is I'll talk to you guys about it. So technically speaking, as we were dealing with Staphylococcus aureus, you shouldn't technically see a yellowish tint. So Staphylococcus epidermidis, however, you do see growth, right, on the MSA plate. And that directs you to understand, okay, is that essentially selecting for or differentiating Staphylococcus epidermidis from other bacteria? What do you guys think? Is it selected for or is it differentiated for? Selected, reason being, I'll just let you guys know why. Because as you recall, it's at 7.5% NaCl, right? If we connect back to the environmental conditions, this is highly concentrated for NaCl within the growth of bacteria. So think about it like this. This is an extreme halophile because essentially it is tolerant to a large spectrum or to a greater concentration of NaCl, meaning that essentially it's still able to grow, and thus it's selected for within this plate. As you can compare though, E. coli, for many of you, is likely not grown, has not grown, correct? Has anyone had a few colonies grow or anything grow for the other two sections? No? Colonies? Are there colonies? Yes or no? No colonies? For yes, for E. coli. For MSA?
Yeah.
Yeah. So then let's go to the next one. I just want you guys to understand this though. There are some components that do make it differentiating, such as the mannitol and the phenol red. When combined, essentially that will help elucidate a yellowish tint, meaning that these bacteria, like Staphylococcus aureus, would essentially be mannitol fermenting bacteria. So the difference between the red and yellow is whether or not they ferment mannitol. The difference between them growing and not growing is whether or not they're able to survive at higher NaCl concentrations or higher osmotic pressures. So think about that. Think about, okay, the salts, the pHs, the temps that were extreme on either edge from the environmental conditions that we explored. If they're able to grow abundantly, then that shows that these bacteria are selected for if used in these plates, ideally. Or at least that they're able to grow at certain conditions. Whereas for differentiating, you have the different metabolites and colors of dyes that would essentially change following fermentation. From in this case, red to yellow, and in the Snyder test, from forest green to yellowish green. So just bear those details in mind, or lemon green actually. So sorry, lemon yellow. From the Snyder test, forest green to lime yellow, sorry, lemon yellow. So bear those details in mind. There's changes in colors to differentiate different properties of bacteria. For the McConkey Agar, you have crystal violet, which you probably remember from gram staining, also for biofilms, which you'll see today. Believe it or not, you have the delectable crystal violet right there in the middle bench. So you guys will be using it for your biofilm. Let me just quickly go over. Do you guys see? Actually, let me know, what do you guys see from your plates? Yes. Okay. Okay. For the Staphylococcus epidermidis, everything okay, Carlos, by the way? Yep. Yeah. Okay. For Staphylococcus epidermidis, do you see any growth? Yeah. No? No? Okay, what do you see? Yes. 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 It does. Actually, let me clarify. What was your point, Connor? We'll talk about that as well. And the answer is yes. So I'll talk about Curtis's point just in a moment. I want you guys to, you can also look here at the, so look at the ingredients, right? Crystal violet is like a dye. Actually, it is a dye, but I do want to emphasize that this differential component is different from neutral red. So, you know, these two different colors, as you can see from the Staphylococcus, from the E. coli. Can I recall, was it E. coli that you had the halo? Which one was the one that was purple? E. coli or Staphylococcus? E. coli. E. coli? Across the board, is everyone having for the McConkey Agar plate? Yeah? Okay, so I just want you guys to understand this. It's a bit interesting, right? Because if your color, is the color pink or is it a hue pink, right? Purple for some of you? I want to see. Okay, do you mind? Okay, perfect. So perfect, this is good. Let me explain. Is E. coli gram positive or negative? Negative. Technically speaking, you should see it as pink. It is a bit hard to tell because the agar itself is purple pink, so it's hard to always discriminate for all plates. But the idea is that if you're able to see this hue with a halo, then it's then you can expect. It's essentially differentiating your bacterium from the rest of the agar plate. So you want to think about this as a differentiating factor, but also connected to thus the other components. Lactose will also be differentiating because if it's this yellow, it's a bit of a gradient, but let's see if there's any specific details that are provided. Yes, so the acid is produced following fermentation, and that's where the neutral red leads to a yellowish. Wait a sec. No. So maybe I'm seeing it differently because I would have expected something else. Yeah. It's not going on this plate. But there is a bit of a tangent to, not tangent, there is something to keep in mind. So lactose fermentation, you're reducing in pH, right? Now, technically, even though it says here, okay, this is pink red, okay. So pretty much for E. coli, and this actually hopefully makes sense for many of you guys, because if you studied a bit about E. coli metabolism following lecture material, then you know that it does metabolize carbohydrates, and lactose is one of them. So essentially, that will give you a promise to know that, okay, E. coli is lactose fermenting and is selected for as being able to grow on bile salts. Bile salts are a selective component. So you want to think about that with the change in color from neutral red to what appears to be a pinkish red, a pinkish hue following crystal violet staining. So that's where you can come to appreciate E. coli looking like this. Just to confirm, Staphylococcus epidermidis, do you have growth or no? No growth, right? Across the board? Okay. So what does that mean? Is it able, so let me ask, is it lactose fermenting? Yes or no? Is it? Yes or no? How do you know? No, from your bacteria, is there, so do you guys see any growth of bacteria? Yes or no? So how do you know? So think about it a bit. Are you guys able to, so you can't see growth, right? So how do you know if it ferments lactose? You technically don't, because it's being selected for by the bile salts. Meaning that essentially only bacteria that would grow here on this plate should be essentially bacteria that are osmotolerant and bacteria that are able to grow on the, with salt included, essentially, right? So you technically wouldn't know if Staphylococcus epidermidis is lactose fermenting until you took it out into a separate plate, or if you did this on a regular LB agar plate, and then essentially you wanted to do a similar apparatus, including crystal violet, lactose, and neutral red without the bile salts. Does that make sense? It would be a combined, it'd be like a pseudo McConkey agar plate, but the idea is that essentially you're removing the bile salts from the media. So it still would be appearing like a reddish tint, but I want you guys to think about this because this is applicable to your lab exam. So think about it. If you have questions, let me know. But I want you guys to think about what you actually see. Is this what you expect? Why or why not? And can you make further interpretations? Yes or no? Why or why not? And any limitations that you have, which I expect for the FLR, by the way, to not only be described, but to show how you would resolve them. Please bear that in mind. So let's say for your FLR, you notice that the lids were not taken out for the first, for the last two plates of E. coli and Staphylococcus epidermidis at time 30 or 30 minutes of UV exposure. What would you say then? You're saying that limitation perhaps is the lids not being, off, right? Or that the lids were taken off and then direct UV exposure was shown on both sides without a mask. So then after you mentioned that, what's next, right? That wouldn't suffice for me because I want you to see what you guys would actually do with that knowledge. You would say thereafter that, okay, following the fact that the lids, the mask was not placed onto your plates with the lids removed, then I would say, one of the ways to resolve this is to actually have the plate or the mask to be on top of your plates, right? And the lid removed accordingly. So you want to bear those details in mind. Like the goal is to resolve your limitations, right? To make sure that you would, essentially comprehensively show to me that you know the material and how you would best apply that knowledge within to outside of the lab. So bear those details in mind. I do appreciate extensions with the limitations. Even if it's 3, sentences, it takes 3 minutes to write. Do so, and I hope it will be something that you'll appreciate down the road. Any questions? Yes. You can. You don't have to, but that is just a suggestion because technically speaking, you would for the figure caption because you have two halves of the plate, right? A and B. So I need to know what does A mean, what does B mean, right? So you can. Do you have limitations? If you have limitations, okay. So if you have limitations, and by the way, once again, try to have like a good number of limitations described. And they don't have to be all of them, the ones I just mentioned right now. But you do have to make mention of the fact that, there could be limitations that you may want to consider improving upon. So describe limitations. It could be the inoculations. It could be cross-contamination. It could be the UV exposure times, the height of the UV lamp. By the way, I'll slow down if you guys want to write these limitations down. Consider you may have time to conclude it into your discussion. I mentioned 5 last week. Let me see if I can remember them. Let's say 3. Even if you have 3, it's okay. but I'll try to see if I can. You have cross-contamination, actually, aseptic technique. Remember, start off with the methods. Think about, even though it's not mentioned in your FLR, think about what you would have done to make things better. Think about the different setups. Think about how you were working as group, how things were either consistent or inconsistent. You want to think about the following thing. Limitations. Aseptic technique. Some of you already recall this, right? Like when it comes to the L rod, for example, right? It was heated up too quickly, it was heated up longer and essentially without letting it cool down, or there was excess ethanol on your L rod, then you're technically essentially killing off bacteria following even prior to UV exposure. So it would essentially overestimate the effects of UV exposure on your bacteria. So that's one component. Actually, let me just separate it into two different points. Because aseptic technique relates to the L rod. I'll just mention it if you guys can help. So it relates to the L rod, but it also refers to, and this is why I was concerned about, and bear in mind that there is things that I'm aware of that perhaps haven't been accounted for in the manual or from Dr. Brunt's instructions. So the concern about essentially having your lid open, right? Even though you're, yes, using it under a UV lamp, you technically have open exposure for other bacteria to go through. And those that are UV resistant to actually go into your plant, seed into your plates. So bear those details in mind. That's the second component. So cross-contamination is the second one. L rod is the first. The third one is inconsistencies. Okay, I want to see if I can write it because it's a bit hard to write. So they're like sub-points, and you can just keep those details in mind. The third one is inconsistencies in the sense of UV exposure. Some of you may have gone a bit shorter in UV exposure times. So you want to bear those details in mind. Your plating technique is the fourth one, right? Because some of you, as you see from the figure itself, it may have been a zigzag, but it would have not been a full spread, right, with the L rod, which is why you can't technically discern if bacteria were killed, let's say, in a masked region, or if it was simply a poor technique of essentially spreading. And it's okay, actually, not to say that bacteria were killed, but it's okay to say that there may have been essentially misspreading of your bacteria onto your plates. So that could thus impact the growth of the bacteria that you would technically see on the A side, the unmasked side, exposed to UV. That's the fourth point. The fifth point is the height of the UV. So that's a separate point, but really in reality, spatially speaking, if just like light, if the exposure to light, like UV light, is too high up, like right here, like on the lamps above, or too down low, then you essentially will have variations, like what I teach for BIOC17, differences in plant growth, perhaps, or in our case, differences in UV exposure. So technically speaking, were those two lamps that both sides were using, were they on the same height? These are things that you want to consider as well. And I think the sixth point would be like other controls. Like think about temperature, think about humidity, think about you can discuss this, the agar media as well, right? So those are things that you want to consider. Those are literally 7 points, and I don't mind if you talk about 3 of them. So once again, cross-contamination with the L rod, that's a separate one with aseptic technique. The third one, yes.
That also be considered a
Yes. Yes, and the 8th point actually is the one that I just mentioned, and that is the lids itself. I didn't mention this, but it is also the fact that you may have turned your plate up, down, sorry, agar above or agar below. So it would have been either inverted or non-inverted. That's a separate point. That's actually my 8th point. And then technically, the 9th is essentially the lid itself, right? Because some of you may have not covered essentially the what do you call your plates essentially following UV exposure. So if they were covered, then you would see less of an effect to UV exposure. And if they weren't, then you would see what you expect, perhaps. And then the other tangent is like the mask side. So maybe some groups weren't able to mask, essentially, as I mentioned for the first limitation. So that's technically 10. 5 of them essentially are like methodological limitations, right? Like maybe, having the plate up or down, the mask present or unpresent, the lid present or unpresent. That also connects to exercise 2, even though you don't have to make direct mention of it in your discussion, but talk about repair mechanisms in the intro, if you can. So that's like, those were like at least 3 points. I mentioned the inconsistencies as well, the depth, the height of the UV, the exposure, even the location of your plates is something you want to bear in mind, right? So think creatively, critically as to what I've described. Think about what your next steps could be. And that way, inshallah, you'll have an amazing, thorough, not just discussion, but FLR. And I try to consider those details in mind when I give you guys your grades. So keep those pointers in mind. If you have questions, let me know. We'll go briefly now to the DNase hydrolysis test. So likely, you guys have 2 colonial growths or just 1? Just 1, and is that of E. coli? No, E. coli? Yeah, perfect. Okay, so what does that mean then? Can someone tell me what they can interpret from that data? And by the way, consistently, all groups have essentially only E. coli shown with a halo. Yes or no? Amr, is that correct? Yeah. Cynthia, no, Fadliha, is that correct? Yeah. Okay, Funmi, is that correct? Okay, Marwa, is that correct? Molina, is that correct? Karis, I know you showed Scott as well. That's correct. And now I want you guys to actually show me your interpretation. Either of you. Maybe both of you. Okay. Yes, and so, okay. Sorry, continue. Okay, that's actually a good point. And I don't think it's been specified, so I'll make mention of it. Scott, was there anything else you want to mention? Same thing. So let me ask, or okay, I'll talk about it. It's okay. But great job, both of you. Let me just relate to some of the ingredients that are shown here as well. It is both a selective and differential media, and the components include 1, methyl green, which ideally would be differential because essentially will show a change in color, and it is mentioned as a cationic stain. Oh, okay. Basic dye. There was other things. Methyl green. You have the nutrients. So okay, you have the basic nutrients that are required for agar to grow. That's incorporated into the DNase plate. You have DNA as well. This is something interesting that you could consider. And then methyl green. So methyl green, I just want to wrap our heads around this because it's a bit different in this regard. You have DNA. DNA is your substrate, just like how we had for lysozyme, you have bacteria. But DNA, in this case, is your substrate. DNase is what you would find in your bacteria. So if it's like E. coli, and you find a halo where essentially your bacteria is deposited, then you would expect from that streak that E. coli has DNase, as mentioned from Karis. Now, the thing about methyl green is that it appears to be, I don't want to say it's selective. There is a bit of nuance. I want to see if there's more. Okay, we'll say that it is selective as a contact. And the reason I bring this up is because it is a dye. But think about methyl. If you guys recall organic chemistry, methyl, like the methyl groups, or like functional groups at the CH3 plus, right? So think about that. Interesting, actually, because, and that's why I was a bit worried about talking about crystal violet, because it's also cationic. So essentially, or cationic, essentially it's like a cation, and the idea is that it binds to, DNA. And so that's where you see the blue greenish hue. But then essentially, the interesting factor is that itself serves both a differential component of color with the selective trait of a basic dye, meaning only alkalophiles, essentially like basophiles, bacteria that are able to grow at basic environments should be able to grow here. It's interesting why B subtilis, we use B subtilis, right? Yeah, actually, no, it does make sense. And that does, so it does make sense in the sense that, think about it like this. Not only that it's, so E. coli is gram-negative one, right? But B subtilis is gram-positive. And the basic dye itself of methyl green is actually going to inhibit the growth of gram-positive bacteria. That's one point. It's also considering the fact of its ability to grow in these conditions. So generally speaking, the cationic presence of the dye inhibits the growth of gram-positive bacteria, which is, as you can see here, selecting for E. coli's growth. Now, the function is thereafter that you know that E. coli, so only gram-negative bacteria could grow here, technically, but E. coli is also shown to have DNase, meaning that it could break down DNA. So think about those implications. If, let's say, you had a bacterial infection, and hypothetically, even though bacteria, like it's a bit nuanced, but nonetheless, think about it like this: it could inhibit DNA replication. It could at essentially the outer regions, right? So following DNA replication, you have DNA expression. Think about the impacts of essentially bacterial growth inhibiting protein synthesis from like mRNA. Transcription from DNA to mRNA, but likely it does, yeah. E. coli likely also has RNase, meaning that it could also perhaps inhibit RNA translation. So just think about the implications, is what I'm saying. You have the selective and differential components of methyl green present. Methyl green essentially selects for gram-positive inhibition of bacteria. And then the last one is its color as a dye. So, and thereafter DNA technically is differential because it's not inhibiting growth of bacteria. Any questions? That may have been a bit long, but let me know if there's questions. Yes. It's differential. Differential in the sense that differential because we're considering that only bacteria that have DNase would be able to show that halo. Other bacteria that can still grow, that are gram-negative, will still grow on that plate, right? But what's selected for is the fact that with the methyl green, only gram-negative bacteria can grow, and what makes it differential, i.e. bacteria that have DNase or don't, is the presence of DNA as a substrate. Yes.
So if I have two gram-negative bacteria,
yes.
the E. coli will show the halo, and the other one, if it doesn't have DNase, it wouldn't show the halo.
That's correct.
That's what.
Yes, and technically that other bacteria could still grow. Hypothetically like this. Actually, is a good example. Bacteria C, which is Staphylococcus epidermidis, technically could still grow. It just would not show this halo. And then relatively speaking, you could see the difference between Staphylococcus aureus with Serratia marcensis. And nonetheless, this is, by the way, a bacteria that we looked at back in the past. So hopefully you guys can appreciate that in due time. Make sure to record these as figures in your table, sorry, in your lab notebooks. Let's go to the biofilm, and then we'll go to the gelatinase test. Let me just see. Oops. Okay, so the biofilm setup, I want you guys to have the Bunsen burners on. The gas is already on. But what I want you guys to do is something very simple. Believe it or not, quite simple for a final lab. So you have two tubes, right? So have a look at your tube. You have one that both shows has biofilm, one that is turbid, and in fact one that we may have to use again constantly because the pellet is quite large. So just bear that in mind. Continue to resuspend, or it gets fully resuspended from the pellet. So just keep doing it, and you'll see it lift up like a halo actually. So just keep going, and then this is your control. I.e. the media that the bacteria was able to grow in. And what you're going to do is then dump both into a. And then after that, let's see what you need to do. It sounds odd, okay? But there's a reason that you're expecting this to have your biofilm. Does everyone recall what a biofilm is? That's a great response because it connects you back to be able to actually really talking about communities. And essentially, that idea is that these bacteria are in like a plethora, right? Like think about your generation time. These are bacteria growing live and feet. But the thing about or something to bear in mind is that they could actually be post-lactates because interestingly, within this finite volume, growing for at least 24 hours, they're actually able to essentially cover. I want to give another control. So essentially, it's you forming. They essentially form a almost like a capsule. It's the glycocalyx. Essentially, it's forming a biofilm, which is essentially excreting or secreting the development of the ECM, extracellular matrix, within this tube. So the idea is, if you were to look down into the tube itself, you have bacteria. Okay, so imagine this is your bacteria. You see a thin film. It would be like a grid filled with peptoglycans, filled with the byproducts and the nutrients that the waste products and the nutrients that bacteria grow in, but also importantly, they would coagulate actually the tube itself. So you will likely see a hue, essentially a plate, a slanted hue around the periphery on the surface of within the tube itself. So it's essentially a way for you guys to compare biofilm generation between a control and bacteria that grow for at least 24 hours, nutrients and all. So once you're done that, once you've cleaned the tubes, because I do want to remind ourselves aseptic technique, you're going to come here, open up the PBS. After you've after you've placed them into the Wescadine, we will on your bench. You essentially rinse twice each of them with PBS. Go up halfway, you don't have to go up more. Do both for each of them twice, and then rinse and place them back inside. Let them air dry for about 2-3 minutes, and then thereafter, I want you to place it into the crucible. So it's going to be your return into pretty much your return back to the crucible. And you're going to be essentially pouring it into here. to both of them for 10 minutes, allowing them to incubate. And then, inshallah, after that, you could place them inside, do your waskedine, rinse again, but with water, and then dry it out. They'll be inverted, so let them dry. And then you'll observe what you see there. Any questions? So 3 minutes we set up is what inside. Put some water to sponge, actually keep it up. Rinse. It's inside, let it dry for about a minute or two, invert it. I recommend letting it dry before, and then thereafter I put the water half should be sufficient, and then pour it half for both for 10 minutes, and then after 10 minutes, place it inside dispose of the additional media, and thereafter let it air dry. Rinse with water, pH2O, which is the blue one that are there, and thereafter let it air dry and prune it, and then prepare it. Yeah. So in the meantime, you can resuspend your bacteria or your tube, and then you guys can place them into the waskedine. I want us just to follow aseptic techniques till the end, so that way, you know, in case there's any level of contamination, especially with a different hue, then we could talk about that in due time. Any questions? Let's try to do this, like this process, even including waiting, in the next about 15 minutes. Let's try to do it. And if there's any questions, let me know. In that meantime, you can complete the remaining portions of your lab results, and we'll talk about TDT as well near the end with some reflections. So you can get started, folks. On your marks, get set, go. And for those that essentially were waiting for me to talk about their lab notebooks, those that I've mentioned by name, they can come by. That's Amro, Cynthia, and Zara. I don't think she's here. But if there's anyone else, even for this week, let me know. I have Jacob, Garvin, Ziyu, and Samar. So just show me your lab notebooks for either week 8 or week 10 from what I've mentioned. Week 10?
Week 8.
Okay.
Week 10.
Let me just see follow up. Also beneficial for me for validation.
Your phone priority. Problems.
So you said something about
Well, that's the pH would be all like the word pH.
so
pH.
what do you think this lab would have preference for?
You know, like the pH effect, like
What other
which one?
for environmental
pH, temperature, and on it perfect.
so that so I can see
Yeah.
the
Yeah, it came back and I had like 2, 5, 8.
you know that's why it's not only just leaving the room, but also leaving the application. Only words that we want to when it comes back.
Yeah.
Anything else?
That's good.
Is there anything you're missing?
For weekdays.
Basic frequency.
Oh yeah, the figure is done. I figured in the comments to add the paragraph as well.
You want to
Yeah.
then NAM 10, just see the I may have missed it, but maybe that's not. Let me just see the grade.
Top 10?
The glyces are. Maybe I just missed it. So this is for this one. You understand what is prepared? For the after question. I want you to think about it. It's really
Kind of question, you want to finish with day? Also, do you want the flowchart for also the spit section or just the standard?
no problem. Do what I mentioned about the post lab as well. Let's see how they do stuff. And remember the alarm that actually
Yeah, I think the UV is.
The one that you
Okay. Thank you. Okay, so is this the
no, I wanted to confirm because some students are giving essentially it would have been incorporated just before the FLR. So I just wanted to follow. But apart from that, you have the thermal death time, you have the environmental conditions, you have leave it on that.
Yeah, I do have the environment.
Do you have the thermal death time?
Thermal death time, so I couldn't make use of it. We're just having it, so I was like,
I adjusted it, so you should see it. I needed it on purpose.
wait, was it not only for Bible count?
What about oh, you're talking about the how about the Bible?
Oh, the Bible counts I used for decimal.
Do you have one?
Oh yeah, that's decimal. That's decimal reduction. So wait, actually, it should be here. That's one.
That was
Okay, that's one. Yes. No, this is for, this is temperature 50, this is temperature 60.
Yes, okay.
So these are the
So then it's just the last component, the intro
for like 89.
Great job. So folks, if you haven't received your generation time assignment, let me know. I could give it to you directly. It may help. Let me just make sure. Yes, that is correct. I just want to know, there was just one, right? There is. I have labeled grades for each person, and I'll discuss it briefly. That's correct.
The T is the grade?
T is total. I'll make mention of a few things. I have scaled the grades. By the way, follow up, folks. I have scaled the grades from what you see on paper. So don't worry if like it was too low. Or even I won't say too low. Like we had an 87% average. That is extremely good. Like that's like a 4.0. So I want you guys to think about like what your next steps are, especially like thinking about the well, not just the FLR to come, but more so what other things you could do to improve. Who's next? Sorry. That's okay. That's okay. Let me just see if I can get everyone else. Smart ones. Were these your three? We just want to make sure. No, no problem. I is for introduction, R is for results. I'll talk about the breakdown. Because I haven't been able to mention it to others, I'll send it on Quirkus, but essentially I have a breakdown. I like to be objective when I give you guys results, but that's where I'm more merciful on giving you guys a bump. The bump was, I think, how much was it? I think it was 0.1 or 0.3. I forgot. But nonetheless, if you have questions, let me know. Are you sure? Okay, this is week 10, I believe. Okay, week 10 is what I have it for. So like this week, pretty much what I gave as reminders.
Oh, okay. Okay, so it's not.
Not from the previous, no, not to my understanding.
That's just to add on to what you guys.
Not just from today, but I think there was something missing from either was the post-lab for week 9 or pre-lab. I think it was a pre-lab for week 10. So intro, flow charts, if you don't have it. If I'm wrong, that it could be something else. But inshallah, I'll have a look at yours. And I'll have Jacob as well. But do both of you, I know some yours, yes. Okay, both of you. But Amro, did you give me yours in person or not? Okay. Did I give you a grade for it in person via email? So just give me a moment. Because some of you, I need to make sure I give everyone theirs. Alta, this is your unknown assignment. No problem. Amazing. You get 2 together. No problem. 2 minutes suffices as well. Yes. Folks, when you do the crystal violet, I would like to be there. I don't want you guys to stain yourselves. It is liquid crystal violet. So, did you guys get stained? Okay, that's good. If you guys didn't get stained, then that's okay. If you are concerned, please let me know and I'll help you. There are some people that have names in another place. Okay, Jacob. Well, and I can answer your questions. I could do it even now if you were in line before that. No problem. The review's online, so I can't actually physically play it. So unless you send them via email, but I would recommend it now as soon as possible. So I'll look at the 3 of yours. And I'm not the same for you. Okay, let me make note of it. Just want to make sure. Because I may have already did it, but I thought I wrote it down somewhere. Okay, cool. So this 2, I'll do this seriously. I'll just put week 10. For crystal violet, no. That is correct.
That's the. I felt so bad. Oh, okay.
It was like 20 questions. 20 operating 3 questions and then 50 multiple choice. That was awesome. I think I did really well. But where? That's okay. It's awkward. You can't leave your house. They bought it. You go 0 flat.
No, it's on the right discussion.
That's my bad.
There are a few discussion questions.
Okay, perfect.
If you could just do those two, so kind of take a minute.
Those are
And then,
please. And that's those.
I think you can still.
so it would be just the three of those, the plates that you have from the past, for UV, the discussion, TDT,
Okay.
and there was one more, discussion question. Not that. I'll come to you. Yes. Jacob, and apparently there'll be Jacob, then Marwa, then you. Marwa was, no, she was here before. But I'll go to Alta after you. Oh, I guess. Okay.
Yeah. Oh, you want?
That's good, that's good. That's week 10, right?
Those from week. Those from today. Check.
Yes. I think I did. All two.
Please wait.
We're not.
So are you done here?
Tables?
And so this is for week 8 or week 10?
It was.
I hope this one. So then you guys know.
Like experiment.
A bit of UV, that's environmental.
This one?
Yes.
Oh, the
You know, all this session.
yeah.
We don't,
No. This is
the
What's the difference? I think about that exam, but I don't know. That's
you hear this? I'm with all the so like comparing the past. Yes. You don't have to know. Because that would technically be for time points and you have a bit confusing. So it's better to do it as a basket realistically. And I have it in super count. Kind of.
Good question. Hey, can you tell me what you failed at, but also why I have so many. I thought you were going to be more for effort. No, yeah, I know. That was 0.4. That was fine. I wasn't ready to use all the marks in there.
Your display. This needs to be sorry, online, because this would have been done on the so there was a few things. This is means this is the thing. But if you go here,
You really look at it, yeah, because you think about it, you did nothing to them, and then like it don't like ready. But I understand, and it's so different.
Meaning that you should actually just
Yeah. Sorry, it's different. It's different now. They're complete all the things, but eventually you would have gotten one, right? But the issue is when you like limit.
Label and then I was okay, I'll label my own. I didn't comprehend or going down it twice.
But how do you do these instruments? Like, I mean, like, how do you know the number? Okay, how many boxes?
You come and five. Maybe it's oh, yeah. Oh, yeah. I sat there.
Is this the same? It's the same, like, when you have the same. Oh, yeah. It's the other thing that we decided. And the line. So technically, these are like
Title. That's what title here.
I don't know. But what is so this is correct. So this would be like now, the habit is essentially a concern because what I was mentioning is that depending on when people flow as a line of you may not actually get the accurate generations. That's why like the slope really matters. But I will start to this because you mentioned that so what we do.
Say that's a line of best fit. I was using the same points. So the calculations line is
Because you want to actually make a line
and then choose the 2 points.
within the line.
It would be the points that you added in. Like when you write, when you're making your line of best fit, you're adding something, right? If you take the 2 points, then wouldn't your values be the same?
Yeah.
It's like this.
Let's say this was hypothetically this is a line. I think we choose points that are like, for example, you may not but let's say this is a point. And this is the point that we choose. This is another point that we want to see from that side. So you do essentially this way.
Because my question is if you're right, if you're taking these points, these are the points that are like you're dropping this line based on the points that we scored, right? Right? So when you're choosing these 2 points, or this point and this point, the values will be the same because your points are the numbers. So just because my curve was different, the actual line, the actual values that we use on the line.
So what I mean is like, if you did this, right? Notice the slope. It'd be like something like this. If I literally try to connect the two, something like this. You see how different the slope is? It's not
The same. The question is values. What values do you have on the slope calculation?
the values are
The ones that
pretty much okay. It is possible to use the curve, but you would actually have to have the would have to intersect with it. But essentially, you only should be using if you should use points from. So the reason I bring this up is it is possible that with the scale itself, if it isn't regular, actually this which is why it could also make sense that you essentially follow through with the curve. right? But the idea is to essentially give us this is how you but what I would do is for the film, that was not for at least for this, I'll supplement and then this one should have this one, let me know. Pretty much, not really enough in farming, but I think we'll talk about that. Don't worry, that's what really happened. You can, just me.
This week. Yes. Yes. This is viable. And this one absorbance.
But it's a
I thought density was absorbance. How much density will absorb?
That's why I think you only had
No, this was absorbent.
so you have results for this?
Yeah.
Just technically speaking, like that would you would assume to be the same.
What are them?
Yeah. Because if I look at the two of them and I just by the acid, variable cell density, same with viable cell.
No, they're different.
That's parameters, they're the same. OD or absorption spring is different. That's the right language. Let me know that. And let me just see, keep it with me for a little bit of time. I realize I don't have much time. Let me just find out how people are. Keep it with you for now. Okay.
We have when it comes to the case study.
Yeah, I heard about this. So if you did, I'll keep it for now. Remind me. Before you leave. Okay. Folks, are we done the biofilm? Okay, I see one group. Okay, these two groups are done. Almost, okay. How are you guys doing? Funnel, how are you guys doing for biofilm? Okay, are you guys done the 10-minute incubation? Which group is not done? 10-minute incubation? You already did it? Okay. So this is a bit of a more daunting task. What I'll ask for now, folks, if you can remain seated, I'll answer questions at the end because I know some people have to leave around 11:50. So yes. Is it urgent or if it's okay? I really appreciate it. So let's go a bit forward. So biofilms, TDT is one of the final exercises before you leave today. And I want you guys to understand the concepts, which is also shown on Lab 9. So we'll go back to that slide in just a moment. That PowerPoint. So I want you guys to recall this. Once again, or actually let's take a moment back. Let's relax a bit. My markers aren't working, so let me just draw on the slide itself. Let me know if you can't see. Okay, so for thermal death time, it is the time it takes for your bacteria to essentially lead to zero, reaches zero essentially. Technically, for your lab exam, it may reach 1 in terms of the actual number of viable bacteria. But just bear that in mind because if you're using a log of 1 is technically exponent of 0. So essentially that means that if you have a log of 1, this is actually going up exponentially as whole numbers. But this essentially value should technically reach the very end of your x-axis to determine TDT.
If you're only shown up to here, it is okay to say 70 minutes is your TDT for this curve. But then your D value is a whole other story because you're essentially going from your live bacteria or from the log, and you're going one down. So you're comparing the distance on the x-axis of a logarithmic decrease or deduction by 1 fold. It went down. So you go down by 1 in log, or 10 times lower in the number of viable bacteria or living bacteria. So in the regular numbers, it would be like 10,000 decreased to 1,000, which reflects 90% of the deduction in your bacterial growth. And so and similarly from log, it's a similar pattern. But what you're doing is that with the 10,000 of living bacteria, you're going down to the curve, then going for interpolation into the x-axis. And like the lysozyme, you're looking at the x-axis, looking at the values, and this is about 26 or 27, we'll say 26.5 minutes. And then you have, we'll say 26 for you guys. And then 1,000, so you're going down 10-fold once again. So 10,000 divided by 10 is 1,000. You're going down from the y-axis, interpolating to the curve, going down to the x-axis, and you see it's around 40 minutes on the dot. So 40 minus 26, what is the D value? Say it out loud. Don't be worried. What's 40 minus 26? 14. 14? 14, that's your D value. So let's think of this in another way. If, let's say this is at 50 degrees, let's say this curve that you see is at 50 degrees. If it's at 60 to 65 degrees, do you expect it to be lower or higher? Sorry, do you expect the slope to be less slanted or more slanted? And I'm doing it on my hand, like facing me, but for you guys, it will look like this, decreasing. So would it be at 65 degrees more slanted or less slanted? More slanted, right? Meaning what then? Would it TDT sound faster, right? And that's under the assumption, by the way, that this is E. coli, right? Or a bacteria that would decrease in growth at higher temperatures. Bear in mind the extremities or the conditions of your bacteria and the characteristics of where they optimally grow, be it a pH, salinity, temp, etc. So now what does that mean? If it increases in slope, is the TDT value lower or higher? Lower? Lower? So now that we know that the slope at 65 degrees is larger or longer, more steeper, does that mean your TDT value is lower or larger? Lower or higher? Lower, 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 lower. And so D value lower or higher than 50 degrees? D value now, is it lower or higher? Lower, because they're both proportional. If you increase if the slant, let's say, goes less, or let's say instead of 50 degrees, it was 37 degrees, or an optimal growth for your E. coli, then inshallah what you'd see is that the slope is less. That reflects a larger TDT. It will take longer for that slope for you guys like this, all the way to reach the x-axis, right? And in turn, because it's also less steep, then it would take a longer period of time for that 90% to actually 90% reduction to occur on the x-axis. Does that make sense? Does that make sense, folks? They're proportional. TDT and D values should both correlate with one another proportionally, which is actually interesting for like mathematical concepts. That's the end of lab 9. So I want you guys to, I posted this on Quirkus, so you guys need to do this and sending it to me by either today or the latest being Monday. And that is essentially your values for the TDT. You can write this down if you need to, unless you want to show them to me today. That's also perfect. You need to show me, and I'll write it down too. But I already have it, I believe. So your plates, the 3 plates, figures, detailed captions. I'll actually repeat it one more time. So for your post lab for this week, you can show it to me either today, that'd be amazing, or by Monday. Post lab, photo, yes, via email. I mean, like technically, you guys need to do this, so it would be a sign for me to know that you guys really want to perform well into this course to the very end if you complete the remaining portions of the lab. So that would include the 3 plates. That includes, and by the way, any outstanding items that you're missing from the past that you need to send me, but the detailed descriptions for figures and tables all across the board of like all previous exercises. If you haven't done that, you need to do it immediately, and you can send them to me. But for this, including this, you have, I think I could write it down actually. We'll write it down. Yes. So, let's go. We have 3 plates, right? McConkey, let's say, DNase, using only numerals, legends. Let's continue. 3 plates, gelatinase test. By the way, did you guys get to see the gelatinase test? Who hasn't seen it? I'll let you guys see it. Don't worry. It'll be a good fun. And then 4th is your PET from the microplate reader, which I sent on purpose, so you don't even have to look. Like, look at my message on purpose, because I sent it as a screenshot, so you guys can use. Look back at lab 9 slides. Essentially, the first 2 rows are 50 degrees. You'll take the essentially absorbance readings from there. The last 2 are for 65 degrees. The first column A, is your blank, so you'll have to correct for the other absorbance readings from B to F, I think. B to F. And those go up from 0 minutes all the way up to 3, 6, like all the way up to 18 or even 20 minutes.
Okay.
And incubation, yes.
You said you could use the Bible count, right?
Yes, Dr. Brett wants you to do the microphone. So you need both. So at least this one I was recommended last, like on Tuesday when it was teaching. So you guys just try to do both. It is a good practice. You don't know if you're going to get a similar question, and a question that perhaps will prepare you doing it now before the lab exam. Start doing it as soon as you can, and that won't take too much time.
The first 2 rows are 50 degrees inside.
That's correct. Yes. From the screenshot that I sent on purpose. Use the screenshot once again. And then the last one is the lysosome, which you also have on my announcement. So have a look at both. Determine the initial stock concentrations of lysozyme. Please bear in mind that you're using units, right? Accordingly, nanograms or micrograms per mil. Make sure, once again, always, guys, like, even for your lab notebooks, I see and I'm able to note attention to detail. I know if like you're putting detail into your lab notebooks, and I see some people that perform extraordinarily well, but I want to make sure everyone is on the same page by following the instructions I've provided, right? Detailed figure captions, detailed table captions, below, above, putting in descriptions in between, even for your FLR, right? Have like, for example, 1 paragraph above talking about the intro to the objective or your aim, then referring to your table or your figure 1 for your plates, right? Then after that, you have your figure for your plates for the UV FLR assignment. You have your caption. After the caption, in your caption, by the way, you always need to have a title, a brief mention of methods, like your condition, the conditions of your apparatus or of your result / figure, or even your table. And then the title is specific with your bacteria, your aim, your result, aim / result, and then the assay, which we hopefully, okay? And then you have, that's for your figure captions. Always have this even for your table captions, and then the summary of your results, which you would expand on in your upcoming paragraph. Please make sure that you separate your results such that you have it start off like with an intro, then you show your figure, then you provide these details in the caption, and then you elaborate on your results thereafter. Don't have, like, essentially have it organized like a magazine, have like figures and tables in between your results. Don't have the results just at the top and leave the rest to the bottom, because it's not only you looking back up and down for like a reviewer, but also it shows the storyline or the background of your FLR, and it improves your format accordingly, which is also considered. So keep those details in mind. This is your post log. Send it to me as soon as you can. The sooner the better. And I'm just going to mention this. I'm doing a review for weeks 1 to 5 next Tuesday from 10 a.m. to 1 p.m. That way, I'm trying to help you guys. You don't like to make sure that you guys have the opportunity to excel. This is next Tuesday. The other one is tentative because apparently some of you guys have like an exam that's the day of. So I'm going to move it to the following Monday. I'll give you the dates afterwards, or even on purpose, so don't worry. It'll be at 10 from 10 a.m. to 1 p.m. I want to give you guys time to prepare. You do have tomorrow with Dr. Brunt, but I want to make sure I can also provide any level of assistance in case you have questions. Have an exam too? Okay, let me think then. Can do
post like recordings of them?
I can, but the thing is, I want to make sure that for those that want to participate and boost their grade, they'll still have an opportunity to do so. Okay? You can let me know, and I could try to accommodate. But the thing is, I want to make sure, like, you don't have, like, you guys want to come up, right, like, to attend and actually, like, review the material with me, right? Especially if you have outstanding questions that maybe I went through a bit quickly. Or if there are things that from the manual you want to clarify, like, oh, did we do this microplate reader result? Did we do it at this temperature? What were the results? Discussion points? Any follow-up questions? Those types of details I want you guys to be well prepped for. So think about those details. Any other questions? How's your biofilm working out? Right? And that's your biofilm, right? So this is pretty much your bacteria, it's secreted products, pretty much on the surface coated within your tube. So hopefully many of you have this. Do all of you have something like this, by the way? You can lift it up if you wish. You can have a look. Selfies. But I just want to make sure you guys understand the concept. Hypothetically, as you probably saw from the slide 7, so I'll just show it. Actually, let me talk about it, so I can save that time. Think about it like this. If you have a higher turbidity of bacteria in your biofilm assay, you expect it to look more purple or less purple. If it increases turbidity. More or less? More, right? It's very simple. So I just want to make sure you guys understand that. And if it was less, then accordingly, believe it or not, it would show a similar differential trait for biofilm for the biofilm assay. So think about the presence or the coloration of crystal violet as the differential characteristic of the assay. And then for those of you that haven't done so, this is what your gelatinase test will look like. If it is if it has gelatinase, an enzyme that we developed, then we look like this. Right? We literally because essentially it would lyse out, it would break down gelatin found in the agar, which thereby would solubilize the media. In contrast, you can do a dairy cream flip, right? And you don't see anything happen to this one. It could be your control, or likely for many of you, of E. coli. Actually, I'm a bit interested to know. Has anyone seen E. coli show positive results? You guys should all look at yours as well. There are former groups that take theirs here and then return them back. And then once you're done as well with the plates, after you've taken photos that are a strong amount, you can place them into the autoclave bin. But I want you guys to have a look, compare the results,
So this is something that you would expect if you search online, B-subtilis, does it have gelatinase or not? What is its function? Think about the implications thereafter. Those are three things that you know, and as I'm speaking, are FLR-worthy topics, FLR-worthy questions, and FLR, in fact, not only FLR-worthy discussion points, but lab exam-worthy content. So think about those concepts. Think about the implications of your observations. Think about what you've observed, what you would have expected, and what are the implications of what you've expected, and any limitations that you could include thereafter. So think about that, have a look, share it with your group, the three reps that you have of your B. subtilis, E. coli, and of your control, which was left without any inoculations. Any questions? No? So I want you guys to do it. Yes, thank you. So great question. The tubes themselves, return them back at the very end. In fact, actually, if you don't mind, in the other groups, I see two tubes that are left here. Tubes. You're done? Perfect. So when you guys are done, I'm going to move them outside. Please place your tubes there. You can place the tubes, maybe just to familiarize yourselves. You can place it back here and I'll move them down there. Biofilm, you actually place it in the same space, same tray. There should be enough space. If there isn't, let me know. And then if there are any questions, let me know. We could go straight to, oh, I forgot to mention, the plates go into the autoclave. After you've taken photos and selfies with your beloved, cherished plates, you can take photos, place them into the autoclave. I recommend not opening up the lid, by the way, because not only the smell, but the aerosols as waste products, you don't want them to be, vaporized or essentially rather, inhaled. So just bear that in mind when you are done. Any questions? Okay. Which effort? Which one?
So do we need to do that? Because one, I just did it based on our group alone, and I recorded that version. The other one, I did it with the can I leave it?
That's okay. If you have everything else included, everything like all bacteria, all toxins are included, then that's okay. But one point is that you want to make sure that you're referring to your table before. Like, remember how I was saying, like, have the figures and tables sandwiched in between your recipes.
Oh, yeah, there'll be a figure here. I will one for, there'll be one here as well before I start discussing my results.
Okay, but technically, the table captions go before.
Okay, I'll write them before this.
Perfect. And folks, I want to make another point. Your captions and your titles for figures, for captions, your title and their descriptions are all incorporated together. So don't have them separated in format. I'm not saying it for your case, but I learned this from, I've seen some people ask me this question. Have them together, have them uniform. And so that way it's not confusing for me to know if your figure caption is part of your paragraph description or if it's actually a header or a closing, like a caption for your figure. Figure captions go below your figure, table captions go above your table. If you combine the caption, you have to combine the table. So technically then just have the descriptions for both. Perfect. Yes. Yeah. Any questions?
I have a question on how to represent the result. So for figure caption,
figure, about the figure caption or the paragraph? Paragraph description is normally just briefly or like see table what exactly is going on. And then following your figure, you have a caption or detail, like the title. One moment. I have a few questions, sorry, but I'll come to you for each one. Number two is the brief methods, the conditions that you have, the DMAS, 8 half, one is math, one is unmasked, and then the third is the summary results, and also make note of units. That's part of the so bear in mind that but you're having a summary results. So once you're done that summary, that concludes the section. You're pretty much you're talking about you have like 3-4 sentences. One sentence, title, and fourth will be a summary results, cross criteria, and cross line. Then your job description, you're going in very depth. So pretty much interpretation. Whether it's not sections. Then describes more about figure. Then you will repeat the same process. All of them will be together. Yeah. Essentially, focus on efficiency. All that way, any other like see wherever you if you want me to I want to give you that's I would say think about don't worry no problem. Before, okay, I'll go to curious and then I'll answer. Was this the question that you had before? Okay. I'm pressing class related. You can be general. I don't have to like
For the limitations component, to focus on what you've done, but the interpretation and the description of the results should all be classified. Yes, that's. And then the limitations, like from what I pretty much talked about, there were like 10 different limitations. Let's talk about 3 of them. And then let's talk about their impact and how you solve it. Perfect. Very options and their descriptions here. And that's okay. No, it's okay. This is great. I'm not saying anything so far about this. I do want to say maybe so this was corrected because this is the second one. But what did matter? Definitely. For context, I also made an adjustment. So see what you find on the most recent announcement before the meeting update to essentially create a corrected value. So that way the result will go. Otherwise, it'd be very hard to know what the people but apart from that, I don't think so. But I don't think I am. No, I'm not missing. But I appreciate you sharing too. Yes, I appreciate it. Anything else? I will exclude that. Actually, Vietnam. Okay, scale, I'll mention it now. No. Oh, so the reason was, I think there was a bit of miscommunication. So it was the most reliable result recently for this way. It is 15 minutes from home, which is essentially can I look back at it afterwards? My mind is like fixated on. Okay, let me just see if I can do it now. Yes, so okay. If I say your other results, and I think based on the other conditions, technically 70, why would 70 be the most reliable? Then since it's technically, the viable proofs are direct or like the direct measurements. The observance is indirect, so we include both living and non-factory. So my concern was if you're accounting for the 70 minutes based on the observance readings, and how do you know if that would be realized? My question was more like it technically isn't the because it's an indirect method. That being said, even if it's indirect, it could be accurate. But the problem is that because there is indeed a overestimation of generation time, that's where it may be a bit. But technically, it should be better. The generation time. Not necessarily. I understand, but I mean the value itself is quite hard. So that's why I was saying that at least in comparing it being reliable results comes in, maybe 7 time. So let me know the results. But it is supplemental. Folks, I'm gonna pause for questions just to briefly go over through. I want to make sure that those that have to leave, I want to make sure I have enough time. But you could stay near the front if you want. I'll go through some reflective practice questions with you guys. So let's do these. Actually, I wanted to save you guys the time to do this. Can I just confirm everyone's done the biofilm? Everyone has essentially taken down the 5 different components I mentioned, right? The 3 plates, gelatinase test, the biofilm, TDT, microplate, which I've already mentioned on the Corpus inbox, the lysozyme assay. Is all of that understood by everyone? No? I heard no from someone. Okay. So what I mentioned was you're making the curve. You see this curve? So you're going to make it in your notebook, and then you're going to essentially interpolate, find where these absorbance readings are, and then go down to find their appropriate concentrations. Well, you're gonna get the answers. You want to get the answers for all 4 of these. The absorbance values are here, 0.4. Not necessarily this. Yes. So that's where you want to essentially. It depends. So divided by 0.1 or divided by 10 to the minus 1, which is multiplying by 10. So it's pretty much you're just multiplying. So multiplying by 10, the absorbance, not the absorbance. So folks, let me say this one more time. For the lysozyme assay, please pay attention, folks. Lysozyme assay, your instructions are to make this curve, draw it out in your lab notebook as a sketch using the points that you see here roughly, and then essentially follow through with the absorbance readings that you see for patient 1 and patient 2. And the idea is that you're going to essentially interpolate those absorbance values, i.e. go from where they're found, like 0.43 here, all the way down to the curve, then down to the x-axis, determine the values, and then based on that, that will be your diluted concentration. Thereafter, multiply by the dilution factor or divide by the dilution factor, which would be essentially either multiplied by 10 or multiplied by 100, and that will give you your stock concentration of lysozyme in the saliva for each patient. So you could do it for both. You could get the mean. I don't mind if you do it separately. Dr. Brunt maybe would want you guys to do it as a mean. So like this is, think about like this, patient 1, you have the absorbance reading, serial dilution, absorbance reading. These are your 2 absorbance readings. Then following this, you have patient 2 brings in a swab, does essentially gets their spec 20 reading at 10 to the minus 1 serial dilution. This is 0.341. And then following another dilution, this is 0.62612. So essentially, the idea is that you're taking these dilutions into account when you're considering your curve and determining the diluted concentrations of lysozyme. Does that answer your questions? Yeah? Yeah? If there's questions, let me know. Try to complete all 5 before Monday. And then, sorry, I'm a bit tight on time. Don't forget a figure caption as well with the units. Even though it's not shown here, put lysozyme concentration, please. Next step, show. Yes. By Monday. You can do it today. The sooner you do it, the better. But I don't want you guys to forget it because that way you can, let me know how you are doing essentially, and if you have any questions regarding those different components as your post lab. So consider it as your post lab, so that way you can complete it as soon as possible. And that way I could consider for those that are a bit tight with timing. One of the questions disappeared. So let's see. I can open this up. Let me go to the top. Any other questions? Any other questions? Yes, Ashwin. I have told students to do this, so do it in your notebook, but we'll talk about it. Well, so technically that was the final component of the lab, because that's so yeah. This doesn't take too long, by the way, to do. Trust me, you guys. I want you to think about what I've discussed thus far. Include it as your, so consider the 6 actually. Let me just add it in. Consider this with your post lab essentially. And we could walk through it briefly, right? Think about the Snyder test. I briefly touched about it today. The differential component, right? It goes from forest green to lemon yellow. Think about the gradient that you find for the biofilms, right? You go from clear to purple, right? So there is a differential component that connects the two of them. So you want to define what the Snyder test is, what does it serve, and the same for biofilms and how they correlate with one another based on the conditions of what they essentially examine or distinguish between bacteria. Then the Snyder test, I think I've mentioned this. I didn't mention it to you guys, so I'll mention it briefly. Okay, there are 3 components. You have, or there are many components, 3 of them that are mentioned or highlighted. You have dextrose, glucose, that is think about it, it's a metabolite. You have bromocresol green, that is a dye. Think about the relationship between following fermentation. When you have fermentation, the color of the dye itself is actually colorized to essentially change and thereby change from the forest green to a lemon yellow hint. So that's where you find the change in color. Then you have the pH. Remember selective conditions. If it's a high temp, if it's a low temp or temperature in degrees or kelvins or Fahrenheit. If it's the salinity, right? If it's too high, like 7.5% for the MSA Mannitol Salt Agar plate, you have the folks, please pay attention. The pH as well, right? If it's too high or too low, in this case for the Snyder test, then you could see how those selective components play in. So hopefully that helps you understand the second question. The third question, this is very simple. You have the plates in front of you. So have a look, think about what you've done, describe that as like maybe discussion points, right? So you can have this as your discussion for the post lab, pretty much, to be included for your submission. And then real life application, right? I talked about this, right? Think about the Snyder test, think about saliva, think about the lysozyme assay, think about pretty much any of the tests that we've talked about, not even just from this week, but from the past. Endospore stain, capsule stain, gram stain, catalase test, cephinase test, or the beta-lactamase test. Many things that we've done beautifully together. And I want you guys to think about this till the very end, within and beyond your lab exam. And then the lysozyme. Do we know why it's more effective? Let me just see from you guys. You guys understand why it would be more effective for lysine gram-positive bacteria? Yes, Garvin.
Yeah, Assalamu would be more effective because gram-positive is very effective like cell walls, using or essentially thermal, or sorry, osmotically regulated by having essentially big walls from essentially first.
So okay, that's a good point. Now, why though? Like, how does lysozyme essentially alter or essentially lyse gram-positive bacteria? I'll save it for actually curious. Don't worry, you made a great response. Great job, Arvin. Yes. And then if she says things that she's missing, relate them to the entire class. And by the way, folks, please pay attention to your group members. I could see if you're spending time on your phone, and I'll be honest with you, that is not acceptable, right? Like I even as an undergrad, if there are reasons that you have to be on your phone, let's say your parents are messaging you, there's an emergent situation, let me know. But if you are just like, going on TikTok, is that really fair to your other group members in your lab, right? So bear that in mind for your own lab performance and for the performance of the entire practical modules. So bear that in mind. So continue, Karis.
Everyone paying attention? Please say just a bit loud. Which linkages? Can you say them one more time? The bonds? Beta. Glucose. Okay, you did say it correctly. I want everyone to be mindful. Like this is important for your even lecture component of the exam. So you definitely need to know the bonds and linkages. Maybe it'll be like 1 or 2 questions, but honestly, you want to run the risk of being between an 84 and an 85, right? So think about that, or even an 89 to a 90. I want you guys to think critically, understand the concepts that are described in lab manual. Garvin, was there anything else you wanted to add in? Yeah? Okay. Everyone is able to hear? Hopefully. And you can look back at the lab manual to see responses to these questions. Now, let me see. Any final questions? Yes.
For the biofilm, questions like those are like for the 10 and
No. Well, technically, they're dilutions from one another. So this is for like patient 1. Row 1 is for patient 1. Row 2 is for patient 2. Yeah, sorry if it's unclear, but essentially these are 2 different patients. You need to get their values and essentially determine the stock undiluted concentrations, and you could take a mean of the 2 to determine the ratio or the concentration of lysozyme in their saliva samples. You could take a mean for both patients, essentially. Both samples for each patient. Any questions? Technically 4 individual ones, and then you'll take the mean for 2 for each of the dilutions. Technically simple. But they're relatively simple. If you have a mean, it's just like add the 2 and divide by 2. Now, before we end off, let me just go briefly once again. Okay, you need to think about this. Any outstanding questions? I'm going to answer for the next 3-ish minutes for the UV FLR. People have asked me about discussion, about results. I just want to make sure that you guys understand it. Start off with introducing the aim, go into reference to figure 1, have a detailed figure caption underneath your figure like this, then describe your results. After that, refer to table 1. Table 1 caption will be above the table. Talk about the details I mentioned here for your caption. After that, describe this is your final component for the results, but describe what you see from the table itself. And essentially, that would be your results section. The discussion is where you're interpreting the data or doing data interpretations. You're talking about the limitations. You first start off with the aim, talking about how you undertook it, then go into your results, summarizing them briefly about the different trends that you saw across bacteria over UV exposure times. Thereafter, you're going to describe if that's what you expected according to what you've already mentioned in your intro, right? So your intro should have talked upon or discussed or described a few important traits of the bacteria, right? Endospore at 7 days versus 24 hours. E. coli versus Staphylococcus epidermidis, gram-positive or gram-negative, etc. Describe those details, what UV is. I've mentioned many things already. That's something I just want to highlight. Make sure in your discussion, after you talk about what you've expected, and if it was expected, why or why not, discuss not just the limitations, but also extensions, right? And I want you guys for both of those final parts and pieces, not just to refer to primary literature, I want you to describe and discuss literally what you're finding. So that includes, like, as I mentioned before, pretty much, you're not only mentioning your limitation, and the same applies for your extension, but you're
you're accounting for its significance, its relevance. So, for example, there are two plates at T30, E. coli and Staphylococcus epidermidis, that don't appear to have anything except for the second plate where there looks to be one major big colony, right? So you want to describe, okay, what could have happened in that plate, right? You're referring to all of the class data for your results and for your discussion, or for your discussion, you're summarizing it, but you need to make mention of your limitations, their impact, and your potential resolutions, or the limitations themselves, the significance of those limitations, and what you would have done to resolve them. So discuss those three for each limitation you have. And if you, I recommend trying to stay within the page limit, your tables, your figures, and I believe you don't have to have a cover page, but your abstract, if I'm not mistaken, is not covered in the page limit. So still put in as much detail as you possibly can. And I believe that's all I can say. Any final questions? No? Let's briefly end on something. The final question. Question number 6. BIOC17. Yes. If you go up like 4.5, that's okay. If you go up to 5, please email me because I need to, yeah. Sorry. That's okay. That's okay. By the way, are we seeing double spaced? Yeah. Okay. Yeah, that's okay. The reason I'm just wondering is because some people may have like 10 pages, so it's a bit too much for me. Not to say I don't mind, but at the same time, like if you're marking 40 people, it's quite a lot. So nonetheless, it's okay if you please email me if there's extenuating questions that you have. But I want to end off briefly for reflections. I want each of you, or like each group, to just mention one beneficial skill that they found in this lab that they would hopefully take out from the lab following C17. So, or even think about like different things and themes that we've talked about. Group 1, Amro, Ziyu, Ashwin, and Rafati. I want you guys to talk about what you learned. Tell me just like 1 minute, or not even 1 minute, just mention a theme or topic that you found beneficial, or a skill. Sorry? Okay. So like streak plating. Okay. Next group. Amro, you wanted to say something else too, right? Okay. The 3 of you. One of you just mentioned something. Okay. Next group. Yes. For like germicides as well. Yes. Garmin, can you give me something?
Think about all the errors.
Errors? But what about the beautiful things, like the results and observations? Okay. You guys are amazing. Keep up the amazing work, you guys. Group 5. Well done, Garvin. Well done, Marwa. 3 of you guys. Need to get something from you guys. Anything. Yes. Staining is a good one. Okay. Yes, Hajar. Inoculating, like with the loops or with the needles. Okay. So you really think about these different topics, considerations, concepts. I want you guys to really think about them. If we talk about streak plating, sometimes maybe one day, like there was a postdoc actually who graduated in PhD and now is like baking. So imagine like you doing crafting certain cakes, similar to what you're doing with a round table, and you're doing spread plating, right? So like it's almost like pottery. That's one angle. Germicides, aseptic technique. they're connected quite readily. And if you're ever planning to do grad school, let me know. And then, you likely undertake similar protocols in the future, standard operating procedures. And then you have errors, right? So errors you have to definitely be mindful of, like limitations that we've discussed. It also can further enhance for the future, for other practicals that you may take in the future or courses as well. Yes.
Yeah, I don't know about 35.
Not necessarily.
That's what we meant in the virtual lab as well.
The thing is, the one thing that we didn't say that was very beneficial is that there are many beneficial bacteria themselves in the gut flora, right? Even on your hands, right? So, even on your hands. Yeah. It's not mentioned, but the reality is it's the case. Some of them have unique interactions that help. For example, like the melanocytes that are on your skin, they interact with your bacteria, the bacteria that are also present. So not all of them are harmful. Some of them also help, support vitamin E absorption or adsorption. So just bear those concepts in mind. There was a few other points. Susceptibility, resistance. Think about that for your lab exam. Antimicrobial susceptibility. Think about the modes of action, so streptomycin, tetracycline, ampicillin, the penicillin family. All of these things are likely related to you guys because many of you probably already know some of these prescriptions, right, from the pharmacy. So think about the impacts that they have after you take them or your loved ones take them and the impact that that has in medicine, in society, in research, and beyond. So with that, subhanakallahumma wa bihamdika nashhadu an la ilaha illa anta nastaghfiruka wa natubu ilayk. I really appreciate your time, folks. We're done 2 minutes early. Well done. Well done, folks. Well done. So now, are there any questions? If you guys want to stay, you can. I don't mind. And if you have any feedback for me, just let me know at any time. And if there were final questions that I missed, anyone that wanted to show me their notebooks, feel free to do so. Oh, if anyone wants to know as well, I mentioned about a few other pointers from the past. So look back into my messages if you have other questions. Take care, folks. Anything else? I think there is one thing I just want to mention. You guys are awesome. Don't give up. Don't give up. You're in your third years, perhaps. So don't give up. Don't feel bummered by anything that me or Dr. Bunt says. Do your best and leave the rest with the best. So that's all I can say. Keep up. Don't worry. Don't give up. Don't give up, folks. You guys are amazing. Okay? Keep it up. Keep it up. Any other?
I just want to ask, your notebook, lab notebook, if I'm missing anything.
You're not.
Oh, okay.
And if your lab notebooks are complete, validate them with me. But generally speaking, most of you have them completed already. So great job.
Thank you.
No problem. No problem. And the scale was adjusted on the marks. For those that are running for the generation time, the FLR.
That's really great.
That's really great. It's bumped up. So just please don't tell people because you don't want to also get in trouble.
I can't emphasize about it. Yes. So,
But I have no idea what
Okay. So take care. You'd be able to finish this, right?
Oh, I thought you could.
Oh, so what is that page?
This was the.
Did you get it? Oh, there's another more. Notebook. That's week 8 or 10?
You said. The question.
Yeah,
So I just.
that should
So
be.
it's just the.
Yes, like the increments as well. But apart from that, that's it.
Hi, I was wondering if you can my lab notebook.
Sorry, can you repeat? What about it again?
If it's complete.
Let me see. There is one. That is the post lab for week 3.
Week 3?
I didn't see it until now. So do you still have your notebook? Yeah. Can I just have a look at it? Yes.
As well. Actually, but one is I like that.
Good. Footnotes, maybe I always refer to captions.
Oh, that means
I don't know if I mentioned footnotes.
okay. And also like for the captions. How detailed should the captions?
Detail, that's pretty much what I mentioned here. Title, brief mention of the methods, and then the results in the caption. It's not like a methods FLR. It's just
There's no methods required. Follow the outline that's posted. No methods.
like in the caption. I'm just letting them know that they have to describe what the conditions are, what they use, the times, and like those
Okay,
details.
only if it's a figure merchant.
Yeah, that's it.
Okay, not in title, a graph and or the table. Okay, please follow what's posted. Okay,
That's what I'm.
okay. What did you mark the generation time experiment at?
On a 4.
Okay, so I'm seeing 4.1s up, I believe, in your class.
No problem. There's reason. Okay.
And that's probably.
If you guys have skills that are greater, then I'll have to do some other alterations.
Did it just say you only increase those that have lower marks than expected?
No, because that wouldn't be fair. That wouldn't be fair for everyone. And some people did essentially more than that was expected. So they like with the scale, if you actually had 100%, you would have been boosted up. So what I'll do is just like account for that, so that way.
So no marks are going down.
They're not going down. I just have to see where I can now put those other marks.
I mean,
You're amazing. That's what I was thinking.
yeah, I think I was a lot.
Maybe it doesn't even have to be the lowest. It could be like because technically all of them would still account for the same way. Exactly. So it's not gonna impact too many things. I just have to do it as soon as possible. Don't worry. Don't worry, you guys. Trust me that you guys are doing amazing. Just please try to attend the review seminar. The first one is Tuesday, 10 a.m. to 1 p.m. Labs 1 to 5. Yeah. It'll be recorded, but you have to let me know in advance, please.
We're not going.
If you can't go for extenuating circumstances, and that's included for yourself and everyone, by the way. I'm just saying because if you want to boost your grade, then that would be a way for you to essentially have that option. If you can't for extenuating circumstances, we'll see how I can accommodate it. Not necessarily, but you can go maybe from like an 80 to an 85.
And you have to be the whole time.
Camera on.
Well, that's a problem then, because so okay. You have the choice to come. But I would recommend you coming not just for review, but to boost your performance. I won't penalize you guys if you can't go, but I can grade you further up if you decide not to. So just consider what you can do and update me if you can't. Thank you. No problem.
Okay. I'm not gonna be able to show up because I'm not gonna mention for recording and sleep.
Do you want like a separate review?
We just like some recording.
So figure. That's your last
That's
one.
my post lab. I don't remember what I did then. This was the discussion. don't know, what are you talking about? I drew them and then I wrote, I just didn't know at that point that we had to actually write fake captions.
Take care. All three
But
of you
I've
take
learned-
care.
I've learned since then,
That's good.
yeah,
Can you just do me one favor? Right, figure one, figure two, figure three.
Okay.
four.
yeah, absolutely.
Yeah.
Can I get the marker?
Yeah, I'll give it to you.
So then everything else is fine.
Before you do that, though, because this is unknown, you should mention what your unknown was. You
I
should,
talk about it in here, but I can write it there.
that would be easy.
Okay,
You need a pen?
no, I have them, but otherwise, everything else is fine.
That should be it, I believe. That's correct.
Yes, thank you.
No problem.
How many years did you guys do a post-op?
I'm not gonna tell you. You just have to do it as soon as possible. Who was first? I don't know. And was any of the two of you also waiting in line?
We were here and then they were here.
Do you guys agree?
I don't know.
I just collected.
So we were just listening.
Okay.
I just know my mom. Because I have everything at the same. I remember one of the, like, I think week 5 or 6, I emailed it to you, something I missed right after.
For week 3, were you here?
No, that was, I sent you a thing for that.
Okay.
But everything else I here.
And you have staphylococcus epidermidis. Can you see the motility test? Well, okay, you have cones for here. Can you put cones for almonds?
Colon?
Yeah.
Oh.
Apart from that, you're good. Sandra, let me confirm.
Me too.
Oh, I actually got you. You're confirmed too.
I'm good.
You're good. And but right? Yes. Okay. You were absent for 2 weeks. Is that correct?
Yeah.
Which one were you not absent for? And maybe you didn't get your.
The thumbs up.
Which one is it? Week 6 or 5?
It was the week after the semester.
Okay, so it was week 6. So that's okay. And you sent it to me. Interesting. And I will be able to look. And I remember, I normally do give thumbs up. So don't worry. Were you for this week? Like week 5? Week 5 was a semester. Can you just quickly see the line? Yeah. Okay.
I'm also not saving.
No problem. No problem. No, it's okay.
No, we skipped the same thing.
No problem. It's okay. It's okay, you guys. It's fine. You're good as well. You're good. And Hannah, I believe you're also good. Well done. No problem.
I'm not missing.
Let me just check once again. But I just want to make sure Hydra, I got everything for you. Yep. So you guys are good. I hope it was indeed. I hope so. Let's see. Well,
I'm not doing something. Oh, I haven't got that.
I could post it online. Yes.
You got figured out
And just try to participate, folks. For the seminars, try to participate. If you're shy, just text in the chat. But I'm going to be mindful. If you're not like showing your screen and you're just playing around, then like you don't want to be asked certain questions, right? So
You have to be participating.
please participate. Like, try to participate. Your robotics? Oh,
Very excited.
okay, okay.
So I'll probably join that with you.
Okay, but let me know. Yeah, email. Just so you might. I appreciate it. No problem, no problem. Take care. Week 6 should, if you haven't maybe.
And so 7.
8%. How much?
Technically, you could use the ratio that Dr. Brown would want it realistically.
I know, I really. Because I'm working every day.
You have it right now?
No, I was in there. I didn't have TV.
The microplate?
No, I'm gonna do Bible class because I did that one for this week. No, I just like can't.
So do what you can.
So, yeah, it's going to take apparently cost for everything.
No, I can't. I worked 3 days in a row and I have this, we'll learn a half a semester's worth of lectures in one day. And then I have to have my exam the next day.
So maybe during that post that exam, are you talking about the lab exam?
No, I'm talking about a different class.
So whenever you are actually free, like let's say it's even 1
Like
hour.
2 weeks from now.
But please remind me, email me because if I have to send it beforehand, or we have to this tell me in advance what you can at least try to do before then. Because there may be a time that I can't.
I see you take the debug and scale it down to it's like a.
Also, I
No.
I have a lot
I'm
of
using the slope of my results. So basically, I'm just taking D value, I'm inverting it, that's my slope. Then I'm going to put that into a linear derived equation. You can just essentially log over and you're looking at the final. That's what you find your initial. Yeah.
Sorry for ignoring you this. Wow.
That's okay. No, it's okay. It's okay. I just need to make sure. Oh, some people left their things. Okay. Okay. I guess I have to adjust it. Okay. Yes. Yes.
I'm just wondering if I did this right. I don't. I'm kind of confused.
Yeah, so the idea is that you can use either the formula showing this the t value, but for the TDT, you technically want to go all the way to the very end of like the x-axis.
Okay.
So that's what you would try to extrapolate. So you're going beyond your line, or your line of best fit, until essentially it reaches the x-axis. So you can hypothetically estimate if you want to also redo it, that's also possible, because essentially this increments would be much smaller, and maybe every 5 minutes to see the true estimate cross absorbance readings where your TDT value is
So the results paragraph is where I talk about result, right? You mentioned, and then discussion is just interpretation. Right, okay. I think I was just confused on where I put the summary results part.
Summary results.
But you
Yeah.
explained it.
Yeah, so this will be part of the caption, and then you're describing further details in the results thereafter for your figures. And yeah. Or sorry, description, and then definition.
Yes. Funnily enough, the microplates, well, also like.
Pretty much saying about what your objective was and how you undertook it. And then thereafter you talk about the figure. I know, but technically, we want to have it like flow well, so that way as you go from one section to another, there's this continuous track or this format where I know, okay, you're trying to introduce the results section to other readers. So, does that make sense? Yeah. Okay. Beginning, yes. In your intro and your title.
Okay. I always looking for this one.
Kind of like the
For figures.
for the figures, it could be short form. I would recommend if you can do full form. It makes it more professional.
And
If there are other questions, even via email, if it's possible.
also, I know you said you wanted to write stuff done.
Yes, so I would say performance wise, participation is very important.
So there's three main things, technically four. Participation, like how participative or participatory they are in the lab, like in meeting and speaking or answering questions. Efficiency in asking questions. Proficiency is like your ability in the lab to perform the tasks correctly, but also like coherently, like as a group, like coordinating tasks accordingly.
So would that be more of a group work? Because I think
Not
everyone.
necessarily. Not necessarily, because I could see if some people like spend time on the phone selling or like literally from the overall group's performance. That's where the individual proficiency comes in. And like their understanding of the material. Like if I were to ask from me to senior level or senior leader now, what are your next steps? If I were to ask you and say, no, what are you about? So that would be like your proficiency. That's in the practical exercises or in the practical itself. Does that make sense, by the way?
Yeah.
And then the next one is technically, I didn't mention it, but punctuality is very important. I didn't mention about like coming in on time, but like punctuality is your proficiency because if you come in late and like essentially students don't know, or like if you don't know what to do, and students have to update because time is lost now for them completing the exercise, or it may just be time that I'm not saying about you, I'm saying for everyone that like punctuality, like coming in on time, sending in assignments on time, asking questions in advance, and preparing well for the lab itself. So that's kind of like preparation, punctuality and preparation.
I know I was late twice, but that was both because of weather.
I understand. I'm not really like this is the second, like this is the minimum, and for one that I haven't really begun to focus on is mainly participation, performance, so proficiency and performance.
Could you be more specific on where I lack in proficiency?
So proficiency. Okay. I would say one example, I can kind of tell that maybe a bit lost in some of the content that you need, like exercises and what the next steps are within your repertoire. There is maybe some on the other side, but instead of one, if like that's not being defined as a concern, tell me that that's defined that you're trying to do your best for things to be going out well. Because I can bear down in mind and I can account for it. It's just know that if it's not being rolled and I can't otherwise essentially. So that's where pretty much the three roles, almost a mix of them all relate to the overall performance. But it's so students strengths can be from 25 and we can transition. The goal was to see this week for upcoming weeks if inshallah can show me that you are great, or not just inclusive, but show me that you can be better, either before or all of them. You want to showcase that so that way within that range, or so that you can go upwards, even with just percentages or beyond. That way, inshallah, and I would expect the same language like everyone else. I mentioned this, I didn't mention it directly, that each person could assume that they're on the lower end, so that way they could push forward towards getting either the higher end of their bracket in a project or beyond it, so that we are like in another level.
And if I participate in the sessions, would I be able to push myself into C2?
Okay. And I want you to see, even via email, send me emails asking questions, verification, show the level of engagement that you want to get, or more importantly, like to deliver what you can. Inshallah ta'ala, show you that you can increase in all of them, and that way it is expected that you'll get a grade, or you can hope for a higher grade, either within that bracket. So let's say hypothetically you're like to get and by the way, the lab notebooks are generally at like 100%, so that will already like essentially account for anything, but I'm just saying for performance overall, if you show participation actively, like let's say 5 times, then actually, like maybe 7 or 8 times from now to the very end, it will show to me that you really are indeed trying to put the effort on time to inshallah go up.
Question in. Okay.
And if you have any questions for as well, I could post for this week.
Right. I was gonna hop on the call that you did last night, but I didn't get a chance to.
It's okay. It's okay. Inshallah, try to do what you can. And by the way, once again, don't compare your grade with us. It's not right to do so. And I say this because each of us have unique circumstances. Each of us have unique grades for our assignments, for our lives, etc. And so, if you want help or if you need guidance, you can ask your friends. It won't, hypothetically down someone has another bracket because at the end of the day, you may increase more than
Okay.
you may get a higher C2 than that, okay? So don't think about think about moving on your you can think of how can I put you in best of ways. Apart from as you perform yourself individually in this lab, try to see try to customize or specialize in building of yourself up in any performance and all of that as much as you can. Does that help?
Lower than higher.
Right now, I will tell you you're around the area, but I believe inshallah you're progressing to the CQ, and I do want you to see how you can improve. Show me that, inshallah ta'ala, you can not only just go above an 85, but go beyond.
Threshold 85.
Threshold is
Okay.
so technically it goes from 80, but I'm not, I'll put you, I'm not only say I'll put you, but right now you're right around. And inshallah ta'ala, if you show, if you maximize, optimize all four, showing them to me, even if you try to do things in advance or if you prep well for the reviews, then inshallah ta'ala will go up beyond the 85.
You need the subject line to be BIOC17.
You don't have to.
Okay.
I mean, if you can remind me that, no, it's okay. You don't have to. Don't worry. I can remember these, inshallah. But if there's any other things, and inshallah, you'll rise beyond your mental.
Thank you.
Thanks. This is week 10. Yes, this is week 10. Also, they put the different it's very important to put the molecular mechanisms that coordination software. Yeah, and also helps you understand the biochemical relationship. Also, EM or EM, sorry. Z1 and Z1. Okay, no problem. That's what the sound is. You understand. You understand. Yeah, it's probably going to be strong. Alright, so continue. You are good to go. Also, I have my thermal breath time if you wanted. That would be amazing. Okay. You calculated it? How? Okay. So, bit of a manual way, but I went and derived the equation of the line manually to the slope. The first step, I just start to put the slope within that time. Initial PFUs, final PFUs. I know. Okay.
Yep, so I did that calculation over here. This is for the 50 degrees. This is for the 65.
And you made a curve
Yeah,
like you solved for. That's actually a good way. I didn't talk about that, but that's another way to actually calculate that. Take care.
I kept them mostly in exact values. I didn't really do that.
No, that's good. That's good.
Okay, yep, so I solved for one or log of one since that's there for the log one. Okay, this is equation of one for 16.
Let me see. Oh, sorry. Continue.
Oh, sorry. This is the equation of one for 50. This is equation of one for 60. This is me solving. This is my x technically. This is the range. So now x is not on this side, but
Understood.
okay. So my log one subtracted. This over. This is my intercept. All over my slope, which is
Yes.
all this what I included here, and I just simply got this time in minutes, 232 32 minutes, converted, that is 3 hours and 3.8 seconds.
What are the units for this?
Units for this is technically the
If it's minutes, I doubt that you're going to get 232 minutes if you solve the answer.
that is CFUs per minute.
I don't think so because, well, it makes sense for say, the units, but I don't think, because if you notice this, if you do the log, yeah, even with a log, I don't think you would actually. I don't think you would actually get that value because it's too large, 232. Like if you do the calculation again, like I'm not deducting, but I just wanted to see if you can actually get a value that's likely towards 40, even 30 minutes. Like 30 to 50 minutes, because technically, if I do this calculation, this is going to be a fraction, and that's going to be a fraction. So at the very max, I'm going with 80 minutes, but this is too much.
But okay, the thermal death time I got for this is.
Did you use the graph, by the way, or did you just use the formula?
No, I just use the formula.
And which, there's another thing, which ODs, that's a viable count or ODs?
Oh, that was viable count.
Okay.
Actually, all of these are viable count.
And was this from the updated sheet that I sent on Workers?
The one you said was corrected?
I sent a corrected one.
Yeah, I think it was.
Because I had a first one that wasn't corrected for 55 to 50 degrees, and I updated it. So it could be your values are not
Like average those, possible.
Is this your second or initial?
That's my, no, that's my second. See, that's 5.
I'm wondering why though you're doing it the other way.
Because the thermal death time is decimal reduction value, the opposite, or inverted.
Both of them would be going with a slope downward, so both would be negative, but you don't technically have to reverse the two because a log of the initial. Oh, I see. Never mind. I was just thinking something else. Another thing though. Oh, okay. Oh, I missed. Okay.
Also, wait, how doesn't it make sense from my thermal? Okay, from my thermal reduction values, you say that's like the time it takes for me to go down 1 log value, yes?
D values. Thermal death time is all the way down to hypothetically
But
1.
my decimal reduction time is like 1 log value, yes?
That's what it is.
Okay, so if I'm starting at like log value 10 to the 7,
Mhm.
and it takes me 30 minutes to get down like 1 log value.
That's interesting.
1 log value of that. Wouldn't it make sense that my values be like very, very low?
That does make sense, but I'm just wondering why is that the case? Why is that the case that your values are that large even for 30 minutes?
I mean, are they not just into basically pasteurization? They're not like doing like hardcore like 100 degrees Celsius like ultra evisceration.
But I'm wondering why Dr. Bunton would suggest us having values that are towards 5 minutes for the
5
D value. Yeah. And that's why it would make sense that technically your ADT would be under 50 minutes, maybe like 35, 40 minutes, 45 even. So that's why I'm thinking, at least, well, hopefully, let's see, actually, okay, I sent as well another one on Quarkus. Did you see that one or no? Well, that was the microplate absorbance reading. So have a look at the microplate.
It's terrible, my God.
Really?
I looked at it and it looks like it was trending up.
No Quarkus, no Quarkus. The most recent message I sent.
Most recent? Today?
Not today. It was 2 or 3 days ago. Maybe 2 days ago. It was the second last one, just before the message of the meeting. That one.
Maybe I have to look for that.
But apart from that, inshallah, I expect that your absorbance values should be pretty good. Like I would say 15 minutes for TDT. And then well, okay, let's imagine the value is 10 minutes or even 15 minutes. You're going by a base of 7, you probably go under 2 hours, right? You see what I mean? Does that make sense?
Sure. I mean, but those aren't my values. I'm like,
I know
what happened.
But we'll see. We'll see how it goes. I'll try out the absorbance readings, see what you can do, because that's technically part of the post lab. So let me know what your results are, and I can make note of that accordingly. Or we'll see how it fares with the values that we use.
Interesting. Okay.
Apart from that, everything else okay?
Also, I was wondering, did I mainly lose marks for the course or lab performance skills and evaluation for the tables I did, or?
Let me have a look. It could be so. So I would say it's like the performance in the sense of like one of the things you said about so there's 2 minor things. Even though bear in mind like the lab grades, for notebooks, they'll compensate accordingly, like the quality. If you have like, let's say hypothetically 100% for completion, then if let's say you're at an 85, for example, you're faring as around 93, 92, right? So I know some people may be like a bit concerned, but generally speaking, you have the lab notebook grade. If you did pretty good, then they'll get rewarded accordingly.
I'm more curious, like where was my losing grades and lab performance on that thing?
So it would be a bit of this in regard to the clarity. So I would say the clarity is one. The other is the proficiency. So I notice that sometimes, and this is like, I'm often like this as well, doing things independently, but in these labs, or in this lab at least, with the exercises are often quite numerous for a finite amount of, like a limited amount of time, I want to make sure that the group is working functionally together, or like at least functionally well proficient. And so I'm keeping in mind students and how they function as a team, independent of, well, that's my hope, of like taking this into account of their performance as a lab, with their individual punctuality, proficiency, and participation. Participation is pretty good, but I would say try to suffice or supplement that with your proficiency. So like I would say, like in that regard, working with the group members to see what they need to do to catch up. Because mashallah, you're pretty much always ahead, right? For your group. But I want to make sure that you guys, like, they could also either benefit or that you could support them when needed. Because otherwise, if you're focusing, let's say, on exercise 4 and they're down on exercise 1 and 2, then someone needs to either help them, either that because there's something's wrong, or the other option is that you're going to compensate working with, let's say, Fatima to complete exercise 3. You know what I mean?
So I'm being deducted for team leadership?
No, it's coordination, proficiency and coordination as a team. I'm not saying that, and by the way, I'm mindful of like independent work, but I'm just bearing in mind as well that, and I probably touched upon this before, teamwork is also important in this regard. Like making sure that the team is not behind, because even though you may be behind, if the rest of them aren't, then the entire group is
what? Isn't it efficient to divvy out work, or like each of us like divide the assignments to ourselves, and so we can be more efficient.
You can, that is possible, but at the same time, I'm noticing like there may be at times a discrepancy or disparity in which like you may be ahead and they may be a bit struggling with certain exercises. And I want to make sure that they are also, you know, like there needs to be a level of coordination from your side to let them know, hey, the Hajra or Fatima or Marwa, to let them know that, okay, where are you guys on right now? How can I help? And like I would hear those things, you know what I mean? And I would be able to say, okay, Carvin, mashallah, is ahead of the game. He's helping them out. That is leadership, but also making sure you're coordinating and troubleshooting where needed, right? And then you could call me, oh, Mohammed, like something's wrong. Maybe one of the tubes broke, or maybe we just want to clarify this outstanding question. You know what I mean? Like those types of questions, you have asked questions, but I'm noticing that there is like a bit of miscoordination. And I want to make sure that, you know, proficiency wise, that you are helping to manage them complete the tasks that you need to as an entire group. Because the group revolves, like the group works as a, you know, if you work as a group with them, they work as a group with you, but you need to make sure as a group member that they are completing tasks, like, essentially. And then if you find there to be a problem, you reach out to me. Like if you find that, let's say, hypothetically, if I'm in your team in the future and you find that, let's say, you're doing stellar well, and you find that the other group members aren't as active, or they're not participating, or they're not like engaging, you're taking on the burden, let me know. I'll put you in another group. And then that way, that other group, if hopefully that group you can coordinate well, and then you'll complete the tasks even. But because even I understand, often working independently, there's a lot of burden, sacrifice that you put in. And I don't want that to be nor forsaken, nor undermined. You know what I mean? Which is why right now your grade is not bad. It's actually towards the, okay, I'll write it down. It's pretty much one of them. So don't think that, you know, like don't think that. But like and this is not to compare, but other TAs may not even like. It's hard to get that out. I'll be honest with you for lab performance, but you can still get more, right? Participating for the review, sending me emails, asking me questions, checking out articles. Tell me what you think about microbiology, you know, and beyond.
I'm not gonna lie. For me, like when you were asking the questions of like what I learned from this class, I'm not gonna lie, this is very hard for me. I can't really answer that honestly. Most of this I've done. I can't lie.
Okay, really?
Yes.
Like before in another course or?
Sort of, yes.
Okay. You want to elaborate?
I actually am from Centennial.
Nice. And is this a joint program that you're taking or?
No. Well, once upon a time it was a joint program until it wasn't.
Is it paramedics?
No. It was a long time ago, way before COVID. They had also a biotechnology program, and it was linked to an old program. I forget what they used to call it over here.
Molecular biology and biotechnology?
No. That was new. Okay, I think when that came in, that's when they disjoined the partnership and UFD was doing their own thing. They used to have like a specialized like microbiology thing they had over here.
Yes.
But like before I could people eligible to like cross over, they enter the program. So like, well, I decided I will finish my college degree over here, you know.
And so you came back here to the university.
Yeah, basically.
Okay. Are you in your third year or what?
Oh yeah. Well, by this standards, yes, I'm in my third.
Okay. And so you're planning to graduate next?
Yeah, ideally.
Okay. So you still have a lot. That's amazing to hear because a lot of experience that you would have gained, I imagine. And that's really good to know that you're already a step ahead of the game. So that's really good. And then so what you could just do is to see if you could, you know, pass another, like see if you could boost your grade. Refer to what I've mentioned: proficiency, punctuality, performance, participation. Include all of those, incorporate them, apply them to the best of your ability. Inshallah, you'll scale up accordingly. You know, that's what I think. How's that sound?
Yeah.
Hmm?
I'll take it into consideration.
Okay.
All right. Yeah. What these values.
But don't worry about the values for now. I wouldn't like. I actually admired the formula that you used. I was happy to use it. Great job.
But Miss Brand said.
No, you want to know why I? Oh, sorry, I don't want to interrupt. Dr. Brent, I think because she had specific values in mind for the viable counts, which is why if it didn't go too low, following 5-minute incubations, it's possible that the slant or the steepness of the slope is too flat, such that it will take a longer period of time for you to reach the TDP values. But apart from that, I admire that you used the formula because hypothetically, if you weren't given the values of other, like if you were just given the table, but without plotting paper, how would you actually find out the TDT? So that's something that is really good, actually.
Okay, interesting.
Great job.
Yeah, thank you. You've been great.
I appreciate it, Garvin. If you have any questions, please let me know. Keep up on these.
You said Tuesday, 1 July?
10 to 1. And that is this upcoming Tuesday, and then that's weeks 1 to 5, yes. And then the following Monday, I think that's the 14th. Then you'll be able to go through, inshallah, weeks 6 to 10. You could even prep before. There's nothing wrong with doing so, but I just wanted to give enough time for you guys to prep for the final exam on the 23rd. So that would be the week after.
Wait, so the lab exam is the 23rd?
Number 6. Okay. It's incorporated with the lecture. So you'll have like, I haven't seen it, but I'm imagining that you have 2 hours to do. I don't know if she's actually fixed the time, but essentially you'll have like a lecture component of the exam and a lab component of the exam. Or like a lab exam.
It will all be like one final.
That's right.
Oh, okay.
Unless she changes it, because I don't think she would actually, but let's see what happens.
Okay, next. All right, that answers everything.
That's amazing. Once again.
All right, wonderful having you. Thank you for your great kindness and hospitality.
No worries, Garvin. No need to shrug or bow because I leave it all to you. I really appreciate, by the way, your endurance. Keep up the amazing stellar work. I also want to highlight it's very hard. It's often hard to find people that are BIPOC in these stand like environments. So
I mean, yeah, you know.
So like keep it up, keep soaring higher than this guy.
All right. Also, you know, always wonderful to have a fellow brother out here. You know,
I appreciate it. At any time, please let me know if you need this. And the same for Carlos as well. Please let me know. Indistinguishable by the way. So if you need any help, like my brother, and even though I can't like necessarily be a formal brother, but or like a
brother from another
brother.
Okay.
You know, like I'll do my best to support you guys wherever.
Very appreciate.
Anything else? No. Keep up the amazing stuff. You can take a photo if you have a phone. Take a photo and make it easy.
Also, no, it was meant to ask you. Why is some essay about and to ask you about? Oh, this did not occur, or this was not relating to like the TDT microplates.
No, this is different for the. Let me just confirm. Yes, this is for the microplate for 50 and 65 degrees, because we were doing them at different time points. If you see Dr. 9, the lab 9 slides, it shows like the microplate reader and how it was arranged.
Okay, so it wasn't like duplicates, or was it
They
duplicates?
were duplicates,
Okay.
actually. So both of them are 50 degrees across time. And like 0 minutes, sorry, there's a blank, that's column 8, and then B is F, rows columns B to F, for the other ladder columns, refer to the different time points going up and increments of 3 to 4.
Okay, well, I was hoping for something else. I was hoping I mistake.
Oh, okay. Don't worry. If you see see my message on Focus or the inbox, hopefully you'll get understanding.
Okay, so I should be looking for like 3 days prior a newer microplate.
No, I already have it on Focus. So So look at the microplate absorbance readings.
I know.
Not in the modules. My second last message, I sent the lab 10 slides to prep for the lesson plan assay with the screenshot of this table. Pretty much.
Oh, it's in the lectures.
Not
Or it's in the labs.
It's in my announcement actually. So I attached it as like a file. I can show you a screenshot of it. If I have it here. It may take a bit of time because I have things that I have. But for Carlos, is there anything that any questions?
2 questions. So you mentioned the caption for the table should be above, not below.
Table above.
Specifically. I was not aware, and I did submit 2 days ago, I think.
Do you want to edit it?
Realistically, I can.
Try to.
Okay. And then you said 3 limitations. I believe Professor runs at 2 in our line, so I did 2 also.
Okay.
And like I've really compacted it.
Okay.
So I don't know if I can fit 3.
That's okay.
That's fine.
I mean, see, the thing is, technically, I'm marking, like, the reason I provide certain guidelines is so that way, hypothetically, if you were to bring this to another lab or like even a postdoc or like a professor, they would be perhaps more appeased with my method because this is like preparing for publication. So, and I've actually reviewed like for, like a journal. So that's something that I want to make sure that you guys are prepped for if you're interested in research, grad school, med school. A lot of these different types of projects and FLRs are noteworthy to what you would actually want to maintain and format. Like how I was making mention of like the results section being sandwiched between figures and tables. So having the paragraphs in between, the paragraphs about. Technically, you're gonna have for each figure and table 1 paragraph before and 1 paragraph after. So like you have results, and it's a bit hard to write in a very short, simple outline, but you have your intro to the results, brief, then you refer to figure 1, for example. Then you have the figure 1. You have captions. After you mention the figure 1 for referral or for reference, you thereafter are describing the results themselves from figure 1. That's going to be your paragraph. From that paragraph, at the very end, the last sentence or a semi-concluding sentence, you're going to refer to table 1. After that, you show table 1 with a caption above. Thereafter, you describe more details about the table. That's concluding the results. So it's like, yeah. Does that make sense, Ashley?
It does.
You could still edit it, and I don't mind.
It's the spacing. That's all.
Because technically, if you leave things as they are, and you just technically move, like you just cut and paste, like that, let's say hypothetically you had results talk about before your figure, like in detail, you just cut and paste, move them down underneath, following the first referral. So like the last sentence that you should have just before to mention the figure, but then you can place all of that data, all the detailed observations just now below figure itself.
Okay. Yeah, just have 2 paragraphs. I have 1 explaining what figure 1 is, the context of it, and then I cut to table 2. I didn't really do too much of a transition. I mean, I did like a transition sentence, but not like a paragraph in between. So what I'm picturing is 1 big paragraph on top of figure 1, transition in between table 1, and then description of table 1 beneath that. That's the result.
Okay. Also, am I understanding? Okay.
So what I have right now.
It's not like, if you want to know how much that could be penalized for, it only be 0.1.
Okay.
Like I try to be understanding, but I also, there's some things that like I've emphasized, like discussion points, that I really want to make sure that you guys get right, because these sections now are worth 3 marks. Unlike the black assay, which was like 2 marks for the results of discussion, this is worth 3 marks. So I'm looking for specific like content in detail in the respective section.
Okay.
That was your first question, or is there any other question?
That was my first question. The second one has to do with the TBT microplate. So we're doing two different lines, one for 50, one for 65. Which table are we looking at though? Is it the summary table?
It's the one that is sent on the messages on the purchase inbox.
You saw one specifically.
Yes, like with the lab 10 slots. It was like either yesterday or the day before.
Okay, and it's the same data that you posted. Because I know you posted the entire like microplate.
Yes, this is more simpler.
Condensed.
It's condensed and it's just one table.
And you sent this 2 days ago?
2 days ago, I believe. It's with, so if you look at your email, you should see inshallah like up files to download, and you'll see the lab 10 slides. Right under that is a screenshot of the table.
Okay, I didn't see that.
That's what you'll see.
That's it. And then the rest is just the graphs. Okay.
And that'll make it easy. And even the license I'm asking. So don't worry.
I'm just worried about the TBT. That's the only, everything else I have a good idea. It's just the microplate. I'm a little
I know.
shaky reading.
We don't have much time to discuss it like in person, but I hope that inshallah the review will go through more. Maybe may even be tough because I'm trying to condense the material for you guys. So let's see. But if you have questions, feel free to ask. That's perfectly okay. And for those that want to benefit, they can also listen in and essentially tune in the recording.
Perfect.
Any other questions, Carlos?
That's all. Right.
Keep up the astounding work once again.
Thank
You're
you
doing
very
great, by the way. You're doing great. So keep it up.
Try my best. Thank you.
If you need any support, or what I recommend for, as I mentioned for Garvin, think about what I've mentioned. Generally speaking, proficiency, punctuality, performance, participation. Participation is one that I want to see you guys active on. I know it's hard, maybe because either it could be myself and speaking too quickly, or like maybe just reiterating certain points in a different way. But it's important that I want to make sure everyone's understanding, so that hypothetically, if Dr. Brunk comes, gives you a lab exam right now, how would you score? You know what I mean?
Yeah.
Like that's like the
Very
first,
fast.
like it would be like the in-person exam. So and I try to be like generous, but at the same time, try to do your best. Leave the rest to us. That's all I can say.
Perfect.
Well done, though.
Thank you.
If there's any questions, all the best.
Of course. Thank you.
Take care, Carlos.
Nice to meet you.
Take care, Carlos. Good morning.
Yes. Or, yes, the microplates. You want to show me?
Oh, so it's the same thing, just like how I mentioned for Carlos. If you want to see it, I can show you. Sorry, I just have a lot of files.
You're probably good.
I just don't want there to be sensitive.
Look away if you would.
So if that's okay. Let me just see. Quickest. Just before the last one I sent yesterday. So just give me one more moment. I think it's this one. No. Could it be? Okay, I'll look on Quarkus. I know I sent it to you guys. Right, I've also sent down the breakdown as well. Let me see if I can switch Quarkus.
I usually read most of it though. I don't think.
I know I sent it somewhere. Another reason why I may not see it here, but you guys may see it, is because I made it as a reply. So I may not see it because I sent the email, but you guys may see it better than me. So let's see. Oh, and she did change it. Oh, for some students. Okay, never mind. Let's see, like if they have an exemption. Formal lab report, no. Inbox. Okay, this is the one I took a photo of anyhow. If you go down, like you guys are gonna see it, and if you can't see it, I'll just resend it on Quarkus separately.
Oh,
This is the one. This is the blank. Actually, let me confirm. I want to see that screenshot. I have it as well.
I think this one is posted, but there's so many like charts that I don't.
This one is different because, or the one that I sent is different because it's only one table. I want to see if I could find it. You may see a certificate somewhere. It didn't show photos, so I don't want to. You're a bit sensitive,
Oh,
so
yeah, I can look away if you like.
I'll let, do you both mind looking away? Yeah.
Okay. Five questions for the should I also include or consider intensity? Is that a valid metric to consider? Lamps?
That's what I said, using UVC versus.
That's good. That's sound. Now you guys can actually look. So you can look as well, Garvin. This is the table I sent. Pretty much column one. I didn't specify details here as well, but column one is essentially your blank. So you just correct all of these values accordingly. This, like apart from that, you could essentially plot, like just think about the guide that I sent for the microplate regeneration time.
4 points.
There are four. So there are these two rows are for the first. So these two are they're all different reps. A and B are for 50 degrees. C and D are for 65
Oh,
degrees.
okay, this makes a lot of sense.
So these two are reps for A and B. Two rows are for 50. The last two are for 65. Column one is for your blank. And so you would blank and correct accordingly.
Take the mean absolute, subtract, and then
Exactly. So you would subtract all of these other ones. But the difference here is that these different columns are for different time points. So this is when I started off with the E. coli at 0 minutes for 55 degrees and 65 degrees. Technically, this is from another TA, but the idea is that the values or the trends remain the same in outline, in that this is 0 minutes, this is 3 minutes, 6, 9, 12, 15, and 18 minutes. So that's what you're going to be plotting across time for the two curves.
oh, so it's not
What
like
the
an added
means.
the absolute, it's all individually corrected.
Oh, wait, why does it look so
That's
different
correct.
than what we had?
This is what I posted.
No, that's so like.
Yeah, because this was a bit, okay, for context, this is a bit more peculiar to understand because this is going from the incubators to the lines. So it's going every 15 minutes all the way to like 4 hours or more, 5 hours technically. And this would be more difficult because you don't know, okay, which of the, perhaps 16 or 20 different tables am I going to take the TDT values for. So the idea is that you would essentially use that single table that I sent you to just go off from. Because technically speaking, it wouldn't be directly into the incubator. It would have been just the first time point, meaning that it would be more representative because the other concern is that if you let them incubate for like 5 hours, are you going to see a reliable reading for the thermal death time? It may take some time, but the reality is that you run the risk of other E. coli growing, right? And so the turbidity will just go up, up, and up. And that's why sometimes you may see slant go upwards or slightly, rather than being like this type of standard curve, they'll make shift up in terms of slope becoming less slanted. So that's where, like, essentially to make it simpler, rather than looking at this time point, which is time point 21, we just go to one of the first few. If this was an anomaly, then you would go to the second one. And honestly, if you just follow from what I've said, it shouldn't be too difficult.
Which replicate should we use? Just one or?
I technically would prefer both. Like you could correct the blanks together. So let's say hypothetically this was our exercise. I could show you guys.
Can I take a mean for both replicates?
Yes.
Do 2 mean lines?
Yes.
Okay.
And let me just show you now, in fact, to make it easier. You take either average, I think in Google Sheets. Okay. Yeah. So we'll do this. And it's actually it's not as difficult as it sounds because look, it just already suggested autofill. Like, so you know what I mean? Like it's doing that now. Oh, that's one other catch. It have to be 91.
So wait, this is the one you used, yeah?
This is the one that I once against estimates. So these are now essentially your values for the 2 reps. Sorry, so for context, we have extra these 2 rows, right? E and F. This is now for 50 degrees, make it simple, and the other 65 degrees.
Can you just subtract after this as well?
So you subtract the blank, right? You can make it separated here, and you just plot accordingly.
It looks like it's still plotting on. It even looks like it's still like trending upward.
Not really, actually, because this is your blank. This is your first time point, and it's going down. See how it's going down from 0.8325 all the way to 0.7335? So the time point, yes, is going slanted less. Like, I mean, it's a subtle decrease, but it's still something that you could use. And similarly and plus when you correct for the blank, the values will look more modest. And then essentially you would do the same repeating for 65 degrees. Something to note though, you may want to bear this in mind. It will probably stop at this last time point at 7 because this is negative technically. You subtract this value by the blank.
Oh, yeah.
Blank is higher. So you could actually, this is pretty nice because then you can determine what your TDT is.
With the negative?
Well, because the plot will
Oh,
actually go
second
past the zero. It'll pass to the x-axis. It'll go down into the y, like down into the negative for the y-axis. Meaning essentially it'll pass through the x-axis. To determine TDT, it'll be quite easier. They'll make it easier. Trust me, when you do the results.
It's the table 1 that we're using. Okay.
Yeah, just this table.
Perfect.
And I don't want to like focus on the FLR if you're still like finish that. Do this on
There's
Monday.
formatting issues. Probably take me 10 minutes and then I'll do this and submit it probably next week.
That would be stellar.
All right.
I appreciate it. Again.
Thank you.
If there's any questions, please let me know. All the best in this life and the next. I really appreciate your help, your support, your ongoing vigilance and awareness of how you can always both improve. These are important traits, you know, even be it as a TA, as a student, as a postdoc, professor, you know, even a faculty member. You want to continue to see how you can best improve and be the best that you can. So with that, really appreciate
your time. Ready.
Any questions?
All right, Carl, so you're still around ready physics. I'm not sure, but is there a difference between intensity and like frequency?
Intensity, frequencies.
If you're thinking about physics, intensity refers to amplitude. So the amplitude of your wave, think of it from the peak to the baseline of your wave. It's hard to draw.
Frequency is the amount of times it hits in a second. So like if it's longer or shorter, and then amplitude would be like how long.
How exactly? How long?
Sorry, no, go ahead. I was just gonna say how much energy is stored in the wave when it.
Wait, should I consider that then? Wait, should I consider it?
Wait, are you saying it for the FLR?
Yeah.
Oh, I thought you were talking about physics course. That's why.
Oh, no. I think it's just like UVC. I found an article comparing like UVB, UVA, and UVC. They all have like different ranges, but UVC is most effective because its range falls closer to the 250 nanometers.
So that's a frequency thing now.
Wavelength.
Wavelength.
Yeah.
Technically frequency in the reverse fashion.
But wait, so I think about intensity as well, like intensity of like the light going from the lamp to the plate.
That is a plausible condition limitation, but that is independent of you using UVC or UVB or UVA, because technically that is the light source itself. Like how, because okay, if hypothetically UVA, B, and C only differed in ranges for their wavelengths, then amplitude would essentially differ the intensity, right? So like hypothetically, you could still have UVC, but within UVC, some amplitudes are higher and lower. So i.e. some of them are scrunched closer together, like compressed in height, making them less intense. Actually, let me remember. I don't know if it's less or more, because there's a bit of a particular, okay, increasing the increasing amplitude does decrease, I think. I have to remember intensity. But nonetheless, they do differ. Intensity-wise, intensity as in the height of your wave, essentially, from the baseline, to frequency, the width, essentially. And then the width essentially influences the strength, per se, or the pitch, like sound, the pitch of your waves for UV, which has more common effects for like mutagenic relations and effects of cancer and other things as well. So think about like the implications of the results as well in that regard. Like the effects of UVC exposure, not just of bacteria, but maybe other specimens and maybe samples. to reduce bacterial infection, perhaps post-surgery and an autopsy.
Potentially, I found an interesting article. I think it was the SOS gene or something. Or
That's cool.
I can't remember. It's S something S gene, E. coli, that it basically activates to gain some UV resistance at a certain point.
That's
I don't remember what it was called.
If you find out like the conditions and like specify those details from the article, that means
Only against UVA, the one, which is the weakest one, but
UVA. Okay, even still, that's even more interesting because you have that level of resistance at a lower wavelength could indicate some things regarding or like could show its potential for resistance at other UV wavelengths.
I was wondering because S. epidermidis, I would have expected like the opposite, where it survived longer, but E. coli kind of straggled along. So I was like, look out.
From what I was looking at, it was quite relative, it was honestly relatively famous.
Pretty close. There were some colonies for E. coli, like some colonies were able to just like get along way longer. I see the majority died off, but there was like some resistance. But I do have to run to class. So.
Oh, I appreciate the time. Seeing a bit later, and honestly,
To be honest.
no problem. Honestly, I wouldn't mind to spend like days talking to you, like hours at least, to talk to you guys. It's if I ever came out in the wrong way, just please let me know and seek forgiveness because I don't want to be someone who's harsh.
You've shown the most care out of most TAs out there. So I fully appreciate that. Grades are irrelevant to how you carry out the lab and you present your care to us. I feel like that's more important to me.
I appreciate it.
The numeric round was purely gateway into the next degrees.
And well, there is a bit of that, but also like the conduct and some class, for example, will be mindful of students that remember remember them, but also the profs that actually remember how those students conducted themselves. Like if they speak in like a solemn tone voice, or if if they email and like email in a unique way, essentially, maybe in like cursive or something like that. Or like demonstrate a level of like interest in the lab. Like I could see from both of you, even though both of you are like angles apart, like in opposite edges from the lab, I can see both of you guys are interested in some of the content. Or at least, yeah, like I could see your level of engagement. I applaud it, or at least I adore it. I really do. So I would say keep it up. Keep up the amazing work to the best of your ability.
Thank you.
No problem. All the best once again. In this life and the next, I say this because I believe in there being a hereafter. So just letting you know that I'm not in the sciences. Not everything is accurate. So, that's a separate point. I want to take you from your next.
Yeah, no, it's okay.
So, once again, really appreciate it.
I appreciate you and your care. And yeah, maybe, I don't know, see each other again in another class.
I hope so. I hope so. Even feel free to email me, please. You guys, I don't mind. Likewise, anyone else, if there's major things that we talk about, I need some advice.
Nice to meet you.
Carlos, maybe I'll see you in another class.
We were in the same class.
Oh, yeah, well, after this year.
Oh, no, one day.
Take care. Oh, so that's a bit separate, but once upon a time. Correct. Okay. I prefer not to talk about it. Because also it's not like, I mean, it's about being grateful, like not everyone gets the same opportunities. And that's why I find that it's important to be grateful, humble, and like trying to support like the community in the best of ways. You know what I mean? No problem, Garden. And I look forward to the best being once again. You have questions? Let me know if you have anything as well. And I noticed some people have left things. Is that your bag? Yeah. Take care, Garvin. Tuesday. Take care. I do see for other classes. So I prefer it. Yeah. Generally, yeah, like I actually, there are different courses, and they have different apparatuses, different content that is shared. So, they're usually, I would say, they have different expectations, contexts.
Mainly the salt. What this question is very cool. We would like to play with the people back here. You know, that would be dangerous. That's what I'm not used to. Okay. You know, if you did see like some other existing schools, or you know, think about the agents and see how we can find. I don't think the way we would probably see like the formally investigate other or different things that you I don't know why. They kind of wish to have a little more skill. I understand. I think I don't understand.
The Q plates. I don't recall Q plates actually, but I just
When you have to create TFA foods, it's been a while since I've done this. It's like for culturing high feed. That's interesting. Yeah, so you potentially kind of sanding them between like two acres. Acres, like or like you like see the high feed go out. Everything is they're very small so yeah, I can be below to that, but not in there, not so bad.
If I understand, there is a bit of like, you know, like we are going to make that you may not find the same bacteria that you may want to explore right now, but maybe you don't know if you understand the different strains that those same bacteria that you get to make them. So now you can have understanding of the metabolized.
Yeah. I mean, the contribution out of
I used to, it is a nice one. It is a nice one, actually.
Yeah.
Kind of wrong. I'm not sure. رب لا شيطان رجيم بسم الله الرحمن الرحيم والعصر إن الإنسان في خسر
الصالحات وتواصوا بالحق وتواصوا بالصبر. جزاكم الله خيرا كثيرا طيبا مباركا فيه. This concludes the in-person algorithm. Assalamu alaikum.